Right. Are we ready? I think we're on time, and I think it's about time to start it. I see a lot of familiar faces. I've seen all of you, I mean, almost like a lot of you on Zoom, which is exciting, amazing. And now I see you guys here in the room. I wanted to introduce myself. My name is Andrey Skripkin. I'm a father of 5-year-old Victoria. She's almost 5. She's 4.5. She has OGT-XLID or OGT-CGG, how it's known in scientific papers. We also have a couple of parents from the United States and from England. So I would like those parents to stand up and just say hi. It's Steve and Sarah. It's Irina over there. She's my wife. And I saw Miranda. Where's Miranda? Oh, Miranda. Okay. Yeah, and here's Miranda as well. Yeah, we've brought together our kids. We've got Victoria. She's somewhere with the daycare. We've got like a normal kid is just, I'm here as well. Okay, that's another story, guys. You know, that's a big story, but not for today. Yes, we acknowledge other kids as well.
I have a question.
Oh,
That this is not the voice of God. I'm Bud Freaks. Um, I wonder if the families could just walk up and so we could see them because we can't on Zoom, and I
oh,
would really appreciate that integration.
oh, of course. Sorry. Yeah, we're coming. Yeah. Hey, Had, that was you, right? I think that was hot.
That was me. Hi.
Okay, it's good. Hey, um,
Okay.
that's
Thank
just the
you.
beginning, guys. It's just the beginning. All right. So I wanted to talk about the structure of the day, guys. In the very beginning, we will not the very beginning. The first part of the day will be the State of the Union. Basically, we'll go through what we know about OGT-XLID. And it's going to be like multiple sessions, 15 minutes, 30-minute sessions, where different speakers will talk about different aspects of this condition. But what is even more important, there's going to be the second part of it, which we called a workshop, a dedicated session where we want to present a critical path of what we would like to achieve, how we see the critical path to treatment. But also what's important is to hear out people in the room and see what you have to contribute. What do you think should be the critical path to treatment for this condition? And it's going to be a workshop, like an open session, and led by Lance, and hopefully we'll all participate as much as possible. I also recognize that it's just half a day, and there was a reason because we have just half a day. It's our first event. So hopefully in future we will have larger sessions, but as a beginning, we'll start with this one. We'll see what are the outcomes, and we'll move forward with bigger events. Presenters, you will have 15 minutes. We'll keep time. We will show you 5 minutes before you end. We will show you like 5 minutes. You've got 5 minutes. So if you're in the middle of your presentation, please try to accelerate. It's important because we don't want to eat the time of other speakers, and we want to have time for everything. The agenda is super packed. I recognize we can not make all of it, but we cannot make not make it. We have to go through it. Um, Zoom participants, please turn off your camera as possible and microphone as possible. Whenever you want to speak, please raise your hand. And Steve, who's on the laptop, he's the admin of the Zoom video. He will put you on. Speakers, when your time comes, we've got 2 speakers. One is Liujiang and one is Sandra. Sorry. Please, whenever time comes, turn on your camera, turn on your microphone, and we'll put you on. I wanted to also extend and say thank you to specifically Lance Wells, who made this event possible, because we were debating how to make it happen, and an event like that just cost a lot of money, but it was a creative idea that we can bundle it with with the bigger conference that is coming up right after that. So and it helped gather like a lot of people in the room prior to this conference. That was a brilliant idea, and thank you, Lance, for helping with that. We've got this room like thanks to Lance, and all these people are also invited by Lance as well. So I appreciate, Lance, your help with that. I wanted to talk about sponsors, a couple of words who also contributed to that event. University of Georgia and CCRC, again, and Lance, who've identified this opportunity. The Foundation Glycosylation and Duncan Webster. Duncan Webster is a parent like some of us here, and he has his personal story, and he created a foundation that helps other parent organization like ours just do things like these. And he was very open. He didn't need anything, any acknowledgments, and he just said, "Hey, Andrey, you're doing like good. It's a good cause. I'm in the same shoes. I'll just help you." And he donated like something to help us cover the cost for this event. Hart Wilcox. It's a creative agency specializing in experiential marketing and events production. And Steve Hill, who's also in the room, he's one of the parents, and Steve is helping, and this company is helping us do this event. So appreciate your help, Steve, and you guys for coming here and just helping with everything here. Lectins Bio and Lori Young. Lori Young is the CEO of the company. Lectins Bio specializes in innovative research tools and services for glycobiology. Lectins Bio aims at accelerating discovery by streamlining glycan analysis through affordable, user-friendly technologies that support scientists at every level of expertise. So these are our sponsors who would like to say thank you for this event. Now I would like to come back how we're doing on time. I think we're good. I would like to say a couple of words about the why we're meeting here from the standpoint of parents. And the goal, as we see it, is to identify the critical path to treatment. And as I see as a parent, the research can take time. And now, after extensive conversations with researchers who just invested time in conversations with me and my family, I understand that not everything is just happens overnight, unfortunately. But still, I thought it's important to just bring this perspective that for us, every minute counts. And that is why identifying what we absolutely need to do to get to the treatment is very important, and how we can accelerate that. In the world where I'm coming from, that's another message which I had. I'm coming from the tech world and from startup world. You know that sometimes, like when you speak with people and with your friends, and they say, "Hey, I need an idea to get to make a startup." And apparently, when you're in the startup world, you know all the ideas, not all, almost all the ideas. The ideas are there. The ideas are there. It's not rocket science. People know, and bigger funds, investment funds, they know the ideas. The problem is the people, the execution. It's really hard to find teams who can deliver, who can make something happen and happen on time. I don't know if that's applicable for the research world, but I hope that we kind of, not we, but I hope that the research society has ideas. It's just who does what. It's just making sure that the execution is there. And this is where the parents community can also probably can help, and just, I don't know, whatever we can do. We're happy here, and we're happy to come and to contribute to this execution. Because I've heard a lot of brilliant ideas from the research community. It's just a matter of like, all right, who does what, when do we do it, and how can we coordinate our efforts? There are a couple of parents who wanted to come, they couldn't come, and we've made a short video from them, which we would like to show on screen right now.
Hello, my name is Ivar, and I'm Eric's father.
My name is Sage Schultz. I am the mom of Zachary Parks and Theodore Parks.
Kellen was diagnosed with OGT-CDG in November of 2021.
We were first diagnosed with OGT-XLID When Zachary, I think he was about 2.5.
We haven't noticed any unusual things in Eric's development within his first 6 months.
When Theodore was about a year old, we did start noticing symptoms with him pretty much right away.
But after that started a marathon of diseases like otitis, and his development has stopped.
We noticed at about 9 months old that he wasn't meeting milestones.
They did not give us very much hope with this diagnosis at first.
They did lots of tests. We didn't find anything.
When we got this OGT diagnosis, then of course emotions and thoughts are very, very different. We can't escape and what will be here with us.
They told us that, Zachary with his diagnosis, he was very likely to be extremely intellectually disabled and really had no hope for him.
Our day-to-day life is very Eric-centric. He needs supervision 24/7.
Impulsivity, emotional regulation, memory issues with sleep.
We believe that in one day there is a treatment for Eric's and others' disease. How it goes, when it goes, we don't know, but until the researchers are working with it, we have a hope.
I do believe that there's hope for a cure and a treatment, probably in my lifetime and my kid's lifetime. I think that you guys are doing amazing work.
We are very, very pleased and grateful for researchers. You are doing really, really great work.
I think a breakthrough for my family would mean a possibility and a chance of living a slightly more normal life for my children.
We hope to see that it will give really good result in one nice day for all the OGT patients.
I also just want to say thank you to the researchers. We are greatly appreciative of all the work that you guys are doing. This is incredibly important to my family. I'm really glad that we have finally connected, with families and researchers who are actually working on this and can give us some hope for the future for this condition. So thank you very much.
With that, I would like to invite on the stage our first speaker, Dr. Jerry Hart. Dr. Jerry Hart is a professor of University of Georgia. Without Dr. Jerry Hart, we would probably not meet today. This is the figure in science, which, well, I cannot say how much I'm grateful for the fact that you've discovered O-GlcNAc. So thank you for that. And please, ladies and gentlemen, meet Dr. Jerry Hart.
It's a very important meeting, and I hope we all learn something from it that's useful for treating this disease. So, clicker here. Need 3 hands. So I'm going to give the the basics. I probably won't use the pointer that much then of O-GlcNAc and OGT. So what is O-GlcNAc? It's the addition of a sugar called N-acetylglucosamine, and all it is it's glucose with an N-acetyl group at the 2 position. There's only 2 enzymes, which is kind of remarkable given that there's over 8,000 substrates that catalyze the cycling of this sugar. Unlike other kinds of glycosylation, which are very complicated, this is not elongated to more complex structures. And also, unlike most other forms of glycosylation, it's located in the cytoplasm, nucleus, and mitochondria. It's present in all multicellular organisms, some bacteria, some protozoa, some fungi, all plants, many viruses. And yeast do this too, but they appear, although it isn't nailed down yet, use a different sugar called mannose. As I said, it's very abundant. It's also competitive with phosphorylation, and I'll talk a little bit more about that. It dynamically cycles on a time scale that can be long or very short, just like phosphorylation. And this cycling is controlled, as I said, by these two enzymes, O-GlcNAc transferase and O-GlcNAcase. So since we discovered this, it's been over 40 years ago, I guess now, there's been about 5,500 papers involved with O-GlcNAc. And basically what they've shown is it's a fundamental mechanism in metabolism that links glucose, amino acid, fatty acid, and nucleotide metabolism through the biosynthesis of UDP-GlcNAc, which is the high energy donor of the modification. And as I said, it has a reciprocal relationship a lot of times with phosphorylation. It can be at the same serine or threonine residue in proteins, and also 80% of the human kinases are O-GlcNAculated, and so far, the 10 or 15 or so that have been studied are regulated by it in some way, so this crosstalk is really extensive. And because of that, it regulates signaling, transcription, translation, mitochondrial function, cytoskeleton, and cell division, pretty much everything the cell does in response to nutrients. And this is mediated, as I said, by these two enzymes, OGT and O-GlcNAcase. I'm not going to talk much about O-GlcNAcase, but that's a key enzyme that removes O-GlcNAc from proteins. And then OGT, which has a catalytic subunit and these TPR repeats. And this is a crystal structure that was originally done by Susan Walker that showed of a human OGT enzyme. And the enzyme consists of these tetratricopeptide or TPR repeats, and these are mainly docking sites for interacting proteins. And so I'm often asked, how does one enzyme glucnacolate 8,000 proteins in a specific way? And it does so primarily not only by the sequence of the protein, but also by the proteins it interacts with. And if you know anything, it works very similar to the way RNA polymerase II does. These TPR repeats are also required for the dimerization of the enzyme, and then it has this catalytic domain that actually catalyzes the enzyme reaction. And these targeting proteins, this doesn't depict the exact TPR they interact with, but it shows that studies have demonstrated there's well over 800 or so of these targeting proteins that interact with OGT and direct it to specific substrates. And we recently have found a way to mimic that in cells, and I have hope for this because what it uses is dual specificity RNA aptamers. You can link OGT to an aptamer that binds a nanomolar sensitivity to the substrate, whatever substrate you want, and you have to put a linker on it so they can interact correctly. And it will specifically increase the O-GlcNAcylation of a single protein without affecting the other proteins in the cell. And given current advances in RNA technology like was used for the COVID vaccines, I think someday we might actually be able to use this kind of technology to target OGT to specific substrates. And now I think, we don't know the answer yet, but it's possible that some of the mutations in XLID are affecting specific interactions of targeting proteins. And if that's true, and you'll hear more about that later, then this is a possible mechanism for dealing with that. And so, as I said, it regulates pretty much everything a cell does. It regulates DNA methylation, DNA repair, transcription, RNA processing, translation, and all kinds of other things. But it's very abundant in the brain, where it modifies tubulin regulatory proteins. It regulates the trafficking of mitochondria from the cell body to the end of the nervous system. It's very abundant in the synapse region of the cell. I'm going to have to use a different pointer. I should have put new batteries in this. And this is from a review article. It just summarizes the functions of OGT in the brain. Development, synaptic transmission, cognitive functions, circadian rhythms, and also low levels in the brain are becoming more and more associated with causes of Alzheimer's and Parkinson's disease, as well as neuroinflammation. So it's very important in the brain to keep the levels of O-GlcNAc high. And as I said, it does a lot of stuff. And about 40% of the brain proteins are GLcNAculated. It's involved in learning and memory. You're going to hear about this, obviously, and also appetite regulation. So just to say a little bit about its localization in neurons, and I have to admit we don't know a lot about what's going on in the brain, but OGT is enriched incredibly in what's called the postsynaptic density, which is a structure in the dendrites. In neurons, the dendrites are the action, that's where all the action takes place. Synapses take place in dendrites. And it turns out that OGT is highly located at the postsynaptic density. And interesting, OGA is highly localized at the presynaptic density. And this strongly suggests that that O-GlcNAc cycling is very important for neurotransmission. And this is a micrograph just showing how it's localized very much in these dendritic spines. And this just shows that it cycles rapidly in the brain. This is a chemical that stimulates nerves, and you can see very rapidly the GLcNAcylation changes. It also is required for the maturation of the spines. And this is a complete knockout. This is not a mutation. This is a complete knockout of the enzyme. And in the absence of OGT, the dendritic spines that are actually where the synapses occur don't develop properly, as indicated in this cartoon. In addition to that, it regulates the trafficking of the main receptors that carry the ion channels. So the AMPA receptors, which are sodium ion channels that mediate synaptic nervous transmission, are also regulating the trafficking of these proteins to the surface of the cell. So it's important for that as well. And also there's a population of neurons, particularly in the excitatory neurons, that are very sensitive to glucose levels, as shown here, and whereas the surrounding neurons are less sensitive. So that you can see a small change in glucose from 1 millimolar to 5 millimolar drastically affects the GLcNAcylation state of these neurons. And in fact, a few years ago, in collaboration with Rick Hugener's lab while we were at Johns Hopkins, we knocked out OGT in excitatory neurons, and I kept telling the student it's just going to kill the neuron. Well, that's not what happened at all. Within 2 to 3 weeks, the knockout mice got very fat and couldn't stop eating. What this means is it's not only a nutrient sensor in every cell in your body, it's also a nutrient sensor controlling your diet as well. It's your satiety. In other words, this mice was always hungry, no matter how much it ate. And so just to summarize this short presentation, O-GlcNAc is very abundant in brain and neurons. It plays a very important role in neurodegenerative diseases, particularly Alzheimer's and Parkinson's. The crosstalk between GlcNAc and phosphorylation are very important for the neuronal functions. As I said, OGT is enriched in the synaptic density, and O-GlcNAc is important for localization of the receptors that mediate nervous transmission. It's also highly localized right in the action where the dendritic spines are located. It's required for appetite regulation. And I think the major take-home message is this is a vast frontier. We're only beginning to scratch the surface of the role of this modification in the brain and in neurons. And we clearly need a lot more people in the field working on this. So with that, I'll stop.
I think we have 4 minutes for questions potentially. So if anyone would like to ask, please go ahead, guys.
Thank you. So this is sort of simple, basic questions about the genetics of the condition. So is this a recessive? Are these de novo mutations? What kind of mutations are found in the patients? Are these, point mutations, deletions?
Well, Lance and Don can probably answer the specifics of that better, but this is an X-linked gene that's located right near the centromere. And it's one of the most highly conserved genes in all of biology. From roundworms or C. elegans to man, it's virtually identical. And so I think the reason for that is this location right near the centromere, you get no recombination going on. So, and it's so conserved that it would predict that mutations almost anywhere are not good. And it's a large protein. It's 110,000 molecular weight.
Right. I think that's it for questions. Thank you, Jerry Hart. Appreciate it. I would like to invite Professor Lance Wells. Dr. Wells is a professor in the University of Georgia, CCRC, and yeah, thanks, Lance, for organizing that. Appreciate it.
Thanks, Andrey. All right, so I think it's most people in this room know Dan's lab and my lab have spent a lot of time working on OGT-XLID IV for the last decade or so. And so we kind of split things up. I'm going to start with the basics, kind of what we know and don't know about OGT-XLID intellectual disability, especially in terms of kind of in cellulo and in vitro. And then I'm going to leave in vivo some of the animal model and leave that to Dan to talk about. So as maybe not the families appreciate, but the field appreciates, right? There are hundreds of CDGs, right? These congenital disorders of glycosylation. And there's actually, right, I think there's even disagreement in our field whether we should call this a CDG, right? I mean, I think Dan and I would actually prefer to call it OGT-XLID intellectual disability because it's not completely clear to us that it's a disease of glycosylation yet, right? We don't have that evidence to make that definitive proof. But regardless of how you want to think about it, right? My lab historically, it's a picture of my lab recently, right? We study congenital disorders of O-glycosylation, whether that be O-monosylation and congenital muscular dystrophy, or whether that be OGT and X-linked intellectual disability. And it actually turns out tomorrow, 10 years ago tomorrow, I was at an SFG meeting, the Society for Glycobiology meeting, and I actually disclosed the first mutation in OGT associated with X-linked intellectual disability, the L254F family. And the reason I did that had nothing to do with me, right? I'm a basic scientist very much, right? I'm not a geneticist. I don't see patients. But Charles Schwartz is a geneticist who does see patients. And Charles Schwartz gave me a call and he said, I heard you know something about this O-GlcNAc transferase. And that's exactly how he said it, right? And we all kind of laughed and like, all right, you're talking about O-GlcNAc? Yeah, I know a little bit about it, right? And so he got us really started on this, and it was really two talented postdocs in my lab, Krithika Vadinathan, who started this work, and then Nithias Selvan actually got her PhD with Dan, and then came over and did some postdoctoral work with me to continue this work. And so this was the first family, right? And soon as you see this family, right, you're like, excellent, right? Really clear. So this one lady, let's switch days. This one lady had 8 children with 3 different husbands. And so you see affected males in actually from all 3 marriages, or I don't know if those are marriages, from all 3 unions. And you see that a lot of the females are carriers. Also, one thing I want to point out here that we still don't understand is the females are X-linked skewed, usually greater than 95%. And that's a lot of skewing for a mild disease, right? And so there has to be something going on very early developmentally to cause that type of skewing, right? And that's something I don't think we fully appreciate yet. And here's the three family members. And so we published this in 2017. Actually, that same year, Dan published a couple other patients, right? And so the list started growing. And so we went from this one mutation to all of a sudden we had 5 mutations. So as soon as Charles and I published this paper, We had a couple clinicians reach out to us with their patients, and we had 5. And they were all in the TPR domain that Jerry talked about, right? So this domain that's involved in protein-protein interaction. And they were actually, initially, these first 5, it was only in 3 or 4 TPRs kind of clumped up together. And so that made us probably run down a road that maybe we should have ran down so hard, right? Which is, we thought all the mutations were probably going to be here, right? And that there was some protein-protein interaction that had been lost, right? And then as time went on, and Dan probably has an even more complete picture of this, is now we have mutations everywhere, right? We have mutations all across the TPRs. We have them in both domains of the catalytic. We even have them in this intervening domain now, right? So there are mutations all across this protein, right? All sorts of variants. And so getting our data together with some of Dan's data and the clinicians that we both collaborate with, we started trying to decide whether it was a non-syndromal or a syndromal disease, right? So non-syndromal intellectual disability would be intellectual disability and there's nothing else there, right? But it turns out that we think it's probably syndromal, right? We see dysmorphic features in 12 of the 13 initial patients we looked at. We see eye abnormalities at a high penetrance and clinodactylism, namely fifth finger clinodactylism at a pretty high penetrance. And so that's probably starting to tell us some things too, right? Because that's starting to sound like Hox genes and things like this maybe, right? Because we also, at least in our L254 patients, they all had hypospadia, right? Which again, starts making you think about things like early developmental Hox genes, maybe things like this, they're not maybe involved, right? And so we know we have a syndrome. And our initial studies were somewhat frustrating, right? So I'm a biochemist by training, right? Actually, I was trained by Judy and then by Jerry, right? And who are both in the room. And so, you know, we took these proteins, we made the variants, and we said, what's wrong, right? And in terms of their kinetic parameters for all the TPR variants, nothing's wrong. It doesn't matter whether you use a peptide or you use a protein substrate, at least for the protein substrates that we used, which might be a caveat, right? There's nothing wrong. You know, are they stable? Most of them are pretty stable, right? There's a subset of variants that impact expression and thermal stability, but the vast majority of them are stable. Dimerization's not impacted for the vast majority, even though we now have a variant that looks like it's got a major dimer problem, right? But for the most part, it forms, you know, a healthy dimer. So there's nothing grossly wrong. There's nothing grossly wrong with the protein. So in cellulose studies have been, right, maybe not surprisingly, all over the place, right? We've used lymphoblasts from patients. Some people have gone the route of iPS cells. We've done a lot of overexpression in cell lines. What my lab is primarily focused in is Cas9 engineering cell lines. So we've made human ES cells. Others have made mouse ES cells. And one of the reasons we did this is with iPS cells, while they would be from the patient, my concern was the phenotype is so subtle, right, that you would never be able to say it's because of that one variant. And so we wanted to go into a homogeneous genetic background. And so we've done Cas9 engineering in human ES cells, and now in some neural cell lines like M17 cells, which is a neuroblastoma cell, where we have about 16 or 17 of the variants in those cell lines. So that's in cellulose studies, right? What people are doing. Well, is there a slide? All right, anyway. OGT, what we know is it interacts with multiple proteins. That the interactome is enriched for nuclear proteins that regulate transcription. The interactome is enriched for proteins associated with intellectual disability. That's great. Some of our X-linked ID TPR variants have lost interactions with certain key proteins. Right, that's work by Naomi. We also know we have a host of catalytic domain variants, right, that Jonathan and Laura are presenting at this meeting. What about OGA interactions, right? Do we have to worry about those maybe being messed up? And what about OGT gene regulation, right? GLYCO and more recently a postdoc in my lab is working on looking at OGT gene expression where we've knocked GFP into the OGT to see if we can change the levels of OGT, right? And so all this work is funded by NICHD, 2 different fellowships to 2 of my students, as well as an R01 to my laboratory, and a long-term collaboration with Charles Schwartz, who is now retired, actually. And so that slide was out of order. That was freaking me out. I was like, I still have some data I need to show. All right, good. Sorry, y'all. Wow. All right. So in cellulose studies, right? So we generated these cells. What can we do with them? So, early on, the first variant, I thought we had a biomarker right off the bat. Lymphoblast of L254 showed reduced OGT levels, right? So it just makes the protein a little bit unstable, maybe not a surprise. But the big surprise was it had this huge reduction in OGA, the other enzyme, right? Which doesn't have any mutations in it, right? And it does this because OGT happens to sit on the OGA promoter, right? Which we were able to show. And we showed that this is all at the level of mRNA. And so I thought, hey, we're just going to be able to follow OGA levels, and we're going to know exactly what's going on with all of these variants. It turns out, to our knowledge, L254F is about the only variant that has reduced OGA levels, right? So it's specific to to L254F. And even when we made these were in lymphoblast. When we made the human ES cells, we lost this effect, right? So there's still the L254F mutation, but the OGA effect was lost, right? Which was really disappointing. So where are we heading, right, with all these in cellulose studies, right? So we need to look at protein-protein interactions, especially for the TPR variants. We need to look at catalytic activity, right? Mutations like T570A, right? They're not binding the sugar nucleotide appropriately, right? So they have KM effects. And one of the things people should not forget about, right, is OGT. While we think of it as an O-GlcNAc transferase, it actually can cleave one protein. It actually is a protease for one protein, HCF1, and so there can be defects in that as well that need to be followed, right? The question is, if you have mutations in TPR or mutations in catalytic variants, most of the patients present identically. You can't sort them based on where their mutation is. And so that suggests to us that there's a downstream common effect, whether we get there by a defect in modifying a protein or interacting with a protein, we end up with a similar downstream effect. And that's our thought. And that's why we're doing a lot of things like generating 18 cell lines in a homogeneous genetic background and doing RNA-seq, ChIP-seq, O-GlcNAc proteomics, and trying to address it that way. Some of our early in cellulose studies, right? So looking at the TPR domains of OGT and just defining the interactome, right, using bioID mass spec approaches, which my lab does a lot of mass spectrometry. And one of the things that really jumps off the page here is when you look at the proteins that interact with OGT using bioID, we identified about 120 proteins, a large section of them are involved, already known to have mutations in those proteins that cause intellectual disability, right? 24 of those proteins cause intellectual disability. And those are the proteins that are interacting already with OGT. And so this got us really excited, right? And if you look at those proteins are primarily involved in transcriptional regulation, right? Oftentimes at the epigenetic level, right? And so our ID interactors regulate gene expression, and they do it all over the place, right? From exon activation to nucleosome remodeling, DNA methylation, transcriptional opening, transcription factors themselves, and even things like export and import of RNA, right? So every single level, O-GlcNAc has already been well identified as being involved, a lot by Jerry Hart's lab and by John Hanover's lab. And so we know that these proteins are involved. We know they're involved in intellectual disability, so transcription to us seems like the right place to look. But we'll talk about more about that later. One of the things we did early on with the first 4 variants we found, right, is first we worked in the L254F lymphoblast because that's all we had. It was the first variant, and we had 4 samples. We had 2 controls, and we had 2 affected. And 2 of the affected, these 2 affected, their brother is the control, And then the other affected is actually, I'm sorry, two of the controls and one of the affected are all brothers. And the other one is the half nephew of all three of them, right? Because of the tree. And so we were terrified to do this, I have to admit. I thought it would segregate by generation, by family, right? The genetics. But it actually sorts completely by disease, right? When you do a hierarchical clustering. And it turns out that the vast majority of genes are not seriously affected. But there's 81 genes going up more than threefold, and there's 54 going down more than threefold, right? And so that was great, right? We started chasing those. And then we got these new variants, and we made human ES cells, so we'd be in the same genetic background. And we have now about 379 genes among these four that are either up or down regulated in the same direction compared to the wild type OGT, right? And so a lot of my lab is focusing in kind of trying to figure out what's going on here transcriptionally, right? Is there a common promoter region, things like that to account for this many genes? So for us, it all comes down to, is there a common downstream impact regardless of the variant, right? Because I think, I'm guessing everybody who gets up here and talks is going to talk about what we need in this field, and it's what we'll talk about later this afternoon, is we got to have a biomarker, right? Because without a biomarker, I don't know how we move forward very efficiently, right? We can do the basic science, but I don't know how we start translating that science without a biomarker. And so with that, I'll just say thank you, and I'll be happy to take any questions.
Great talk, Lance. Question about your stem cells. Like you, we see different effects on downregulation of OGA with different variants. Yeah. To what extent do you think that the 20 millimolar glucose that we put in our culture, which is a readout of O-GlcNAc, masks effects?
Yeah. So it's a great question. One of the things we benefit from at the CCRC, it happens to be the building right next to us, is called the Center for Molecular Medicine. And everyone in that building is a stem cell biologist. And so Jerry's lab, actually working with one of them, figured out a model for making neurons and putting them into physiological glucose. Now, the question is, though, we differentiate in 20 millimolar, right? And can we get differentiation to occur in physiological millimolar concentrations? I don't know the answer to that. It's something we definitely need to try. But once we make neurons, we can shift them down and put them in physiological, right? Even take them down to 2 millimolar if we want to, right? But we can keep them at 4 or 5 millimolar, which cerebral spinal fluid, that's pretty good. One more? Stephanie.
Yeah, I was wondering, by all the cell lines that you did, do you have female cell lines as well, or only male cell lines?
So right now, the cell line we chose to use was a male cell line. Even though, 5 of the 18 mutants we have are actually female patients, right? And so we haven't gone back and made those, and it's just a matter of scale, right? And the problem is we're going to have to switch cell lines, right? I don't have a M17 female, even though we can use SHSY5Ys, which are female, right? And we can do it there, right? And so, which is also a neuroblastoma, widely used in the neuro field, right? So we could do it that way. And with ES cells, it'd actually be much easier to use females, right? Most ES cell lines are female, right? We had to really hunt to get the RUES ones, which are.
We've got 2 questions from our Zoom participants, Michael and Hudson. Michael was the first. I will ask, can you guys introduce yourselves before asking a question so that people on Zoom can identify who's asking this question? And let's it be blitz questions and answers, please, because we're at time. Michael, take it away, please.
So I'm Mike Lawler. I'm a professor of pathology at Medical College of Wisconsin, and I'm CEO of a commercial laboratory called Diverge Translational Science Laboratory. I participated in about 6 or 7 gene therapy translations from the bench to the bedside. So I think that's why they asked me here. I don't know anything about OGT. Just a quick question. You had a map of all of the different variants. Are there any of those variants that are, or a cluster of those variants that actually corresponds to a significant proportion, 80% or something like that of the total patient population, or is this kind of spread all over the place?
Yeah, so it's a great question, right? So this is not CFTR, right? Where delta F570 or whatever it is, it counts for 95. To our knowledge, and I don't know, Dan can correct me if I'm wrong, I don't know of any family that once you go to another family has the same mutation, right? Everyone's genetically related, tightly within, all the L254Fs, right, are all there in that one family. Same thing with every other mutation. So we haven't seen any, I haven't seen, I don't know if anyone else has, I haven't seen any example of the same variant in an unrelated family, unfortunately.
Okay,
yeah, which makes
thanks.
life tough, right? So.
It does. It makes it a lot less easy to target a whole bunch of people at once.
Right. Well, I mean, I guess the question, and I mean, this maybe is for a little bit later on, is the question is, will wild type fix everyone, right? And we don't know the answer to that, right? I don't think that answer is no, right?
Yeah, but what you
fix, right? What are we fixing? Right?
And based on what you've said already, you have one mutation where it's sitting on top of another gene, so throwing wild type in may not help for those, at least that subset of them. But yeah, no, it'll be interesting to talk about in the workshop.
Yeah, thanks for the question. Hey, Hud.
Yeah, hi Lance. I'm Hud Freeze, and I've worked around CDGs of various sorts for about 30 years. And we probably ended up discovering 25 to 30 somewhere in there, collaborated on it. So I've had a lot of background in this, and Lance, I think you make a really important point about the physiological conditions. This is something that we've always pushed pretty hard for, and I know most people do 20, 25 millimolar glucose, and of course, that's not physiological. In fact, I've heard that physiologically in the brain, it's closer to 2 millimolar glucose. But we also know that what happens is that the composition of the medium is very important, and that can be changed, especially in the glutamine concentration, to be more physiological. And the other thing is even the stiffness of the media, of the substrate that you're on. So I think all of those things are going to come in to be important, and at this point we don't know which one, but I really want to make a point that the sugar concentration really has to be dealt with, and it looks like you have.
Yeah, I mean, I think you bring up a great point, right? And the problem also is these cells have been cultured in high glutamine, high glucose for a long time, both of which feed into the hexosamine biosynthetic pathway, and
yes,
probably adopted mutations to deal with that, and that's a major problem, right? So these aren't wild type cells. So maybe things are going to be better when we start talking about living organisms, which helps me move to the next topic.
Thank you, Hudson. Hey, thank you, Lance. I would like to invite Professor Daan van Aalten. Daan is the professor of Aarhus University in Denmark, Department of Molecular Biology and Genetics. And Daan is leading one of the largest labs dedicated and focused solely on OGT-XLID. And Daan brought 5 teams from his lab. So thank you for coming, and please welcome on stage.
Great, thank you very much, Lance. And I really like to acknowledge the 4 parents that have organized all of us getting together here. Just to point out that all the AV equipment has been transported from the UK in suitcases. It's not provided by the hotel. It's just done by the parents. So I think this is really amazing. So yes, OGT intellectual disability or CDG. So we've known for a very, very long time that this modification is absolutely essential for development. And this is kind of what we got interested in about 15 years ago. So we know that it's lethal if you knock it out in stem cells. Flies also end up dead at the end of development with very strange homeotic transformations. Zebrafish have a truncated body axis if you knock it down with morpholinos. And in mice, we can't make a mouse that lacks OGA or OGT completely. We can make tissue-specific knockouts, and Jerry gave a beautiful example of that. But even knocking out in specific groups of cells tends to result in a dead mouse. And so for me, like for Lance, this kind of changed when I was approached by clinicians with 2 different mutations than what Lance has been working on, where there were missense variants in OGT that led to a disease. So OGT-CDG, this is a paper together with Lance where we combined all of the variants that we knew of at that stage. And as was discussed earlier, and I think this is very important to make clear, this is an X-linked recessive disease, and all the variants that we know of are de novo. So they are not transmitted. You won't find them in terms of frequency in NOMAD. We have talked to many geneticists because obviously one key number is important here for attracting funding for this, which is the frequency in terms of the number per live births. So we calculate this to be around 1 in 15,000 or maybe even more. So there's intellectual disability, developmental delay, and then we were sort of encouraged to call this OGT-CDG, implying that it's a disease of glycosylation, but that remains to be seen. So at this point in time, it's important, I think, to dial back and to think about what is it that might cause this disease mechanistically. And a priori, there's 5 that we can think of as a lab, which is that variants in OGT affect the expression of OGT or OGA. And the obvious one, which is that loss of OGT activity, particularly catalytic variants, leads to a loss of protein O-GlcNAcylation, and that then leads to downstream effects. Lance already mentioned HCF1 cleavage. HCF1 is an intellectual disability protein itself. So missense variants in HCF1 leads to intellectual disability. This is obviously a very important target. We can't exclude that these sort of misfolded variants of OGT might aggregate, and aggregated proteins are particularly destructive in neurons. And as Lance also said, the OGT interactome. OGT interacts with over 800 proteins, and maybe it's the destruction of these interactions that causes this disease. Now, the scary thing is, at this point in time, we can't exclude any of these 5 very different hypotheses. Moreover, it's quite possible that they all 5 contribute, and to make it even more complicated, it's quite possible that they contribute to different effects, extent for all the different variants. But I think at this stage, the most important thing is that we can't say that it's a disease of misglycosylation. It's a very possible hypothesis and one that we're working on, but all of these are possible. Then to think about the brain, and there too, there are essentially grossly 3 different possible mechanisms that could explain this disease. One is the sort of the developmental one, that is, during embryogenesis, something goes wrong in the development of the growth structure of the brain. The second one is that after birth, synaptogenesis and pruning is affected, so the building of neuronal circuits. And the third one is that it could theoretically be that brain and the synaptogenesis and pruning is completely normal, but it's actual synaptic transmission that is affected. And again, we haven't excluded any of these. It's possible that all 3 play a role, and it's possible that all 3 play a role to a different extent in the different variants. These ideas have very, very severe implications for possible treatments, and I guess that's why we're here together talking. If this was a purely developmental disorder leading to defects in brain structure, there is pretty much no chance of correcting for this in the case of de novo variants where we don't know whether these kids would have these new variants. If it's one of synaptogenesis and pruning, there is a chance that we can maybe intervene with compounds that work through any of these or one of these specific 5 mechanisms. If it's a disease that would be purely due to defects in synaptic transmission, it's very likely that we'll be able to come up with a way of treating this disease. Maybe with, as what was discussed, is gene therapy with AAVs, or with O-GlcNAc inhibitors or metabolic interventions. But the most important thing for us is to figure out which of these 15 combinations, right, 5 times 3, actually is the main driver of this disease, if it's even possible to detect that. And I'd like to ask Steve to really give me my 5-minute sign, because the last couple of slides are the most important. So as a lab, this is what we are focusing on. We're focusing on mechanisms dealing with OGA and OGT expression, and also hypo-O-GlcNAcylation. And at this point in time, we are quite open as to what is actually happening in the brain. We are doing this in stem cells, in flies, and in mice. And I'm not going to talk about these two very much today. And hopefully at the end, I'll tell you a little bit about our mouse work. So this is an example of a family. This family is interesting from a number of reasons. First of all, this is probably a de novo mutation that was generated about 150 years ago. It ended up with a mom who then gave rise to 4 boys. You can see those 4 boys here. One has the wild type cysteine at this position, is completely normal. Then we have a twin and another sibling that have a tyrosine, and they are very severely affected. This family is interesting because these were born in 1966 and therefore are now grossly healthy adults. So they're functioning normally. They are sort of working in a supported environment in Denmark, and I think some of the L2F patients are also sort of now in their 50s or something like that, right? So they have intellectual disability, language problems, autistic features, dysmorphic features that you can see clearly in their face, some skeletal defects. But the reason why they ended up in the clinic was seizures. So they have epilepsy, which they developed in their 40s. So this is one of these variants that is right in the active center. So we can see the cysteine sitting here that is very, very close to the UDP binding site. So having a much larger tyrosine there you would predict would affect catalysis. And indeed, that is what we see. So this is an enzyme assay where we look at incorporation of GLcNAc right at the bottom. So with this variant, we don't see, well, there's actually a tiny band here that you can't see, but is there. So it's not completely catalytically dead. And in fact, it would be impossible for it to be catalytically dead, because then you wouldn't have a viable human being. We can make stem cells like Lance. We try to do this because it gives us an isogenic background. So in this, we can see a clear reduction in protein O-GlcNAcylation, an increase in OGT. And we are now working on trying to define the symptoms associated with this syndrome more accurately. We now actually have over 70 OGT-CDG variants, the majority of which are not yet published. And that then allows us to make tables like this, where we look at all the different symptoms and we look at the penetrance of these symptoms. So intellectual disability, 100%, developmental delay. We can see microcephaly, this sort of dysmorphic face. We are also starting to see some degree of mild heart abnormalities that we are characterizing. So this is then the sort of our picture of the current state of variants, and as Lance already pointed out, they are spread out completely over the gene structure, covering the active site as well as the TPRs. So one challenge is how do you now sort of diagnose, confirm, or dissect new variants. We get an email from a clinician about once every week now. We've developed an assay to do that. Actually, I'm going to skip that because I want to get to the mouse work. But there is maybe a poster today or at the big conference to discuss that. But just extremely briefly, with this assay, we can, without using any patient materials, dissect nomad healthy variants away from disease variants in a stem cell model very, very rapidly. And if there's new variants that you would like to analyze, please come and talk to us. This is under revision at a journal and hopefully will come out sometime in the next few months. Now, animal models. So we have been able to generate animal models for 3 of these OGT-CDG variants. And for us, actually, the fact that we were able to get these mice was a big surprise because I've previously invested $250,000 trying to make hypermorphic OGT mice and failed miserably. So these mice are alive, they are smaller, they have something weird going on with their metabolism in that they have much lower fat mass, which is maybe something for somebody else to look at. Using cohorts of 15 wild-type mice and 15 mutant mice, we are now doing different types of behavioral screening, terminated by tissue collection for MRI and micro-CT to look at the skull and the brain structure. So this is just looking in the brain as a whole, and here we can see what Lance also highlighted, which is that basically nature, and this happens in the brain of all of these 3 variants, very significantly downregulates OGA, both at the protein and the mRNA level, and it does that to rescue O-GlcNAc homeostasis. Because this is a catalytic variant, so there's less GlcNAc, and there's this beautiful system which has been described already 20 years ago that links levels of O-GlcNAc and OGA and OGT to each other. Some of these mice are hyperactive. Oops. So you can see here, this is an open field assay, the C2Y mice, so this is with the group of patients I showed earlier. They are hyperactive, they're running around in their open field like mad. But this is not true for all variants. So this other variant, the N2Y variant, it doesn't do that. If we look at the sort of hand-eye coordination motor skills of these mice, so this is an assay where we ask a mouse to step onto a bar and to go from one end to the other. I'm not actually showing any stats here, but I thought I'd show the video because that's much more illustrative. So you put these mice on the bar and you ask them to go to the end so they can climb down. You have the wild type on the left and the mutant on the right. And you can see very clearly that this mutant mice has problems with coordinating with the hind paws to be able to keep onto this bar. This is clearer. So here we have a horizontal bar. We're facing the mouse, facing away from the end of the bar. It first has to turn around 180 degrees and then go to the end. And you will see, again, wild type on the left, mutant on the right, that the mutant mice is kind of not able to do this 180-degree turn, and then in the end actually has to be rescued before it falls off again. We're doing some learning and memory assays. The simplest of that is the T-maze, where normal mice make sort of this left, right, left sequence every time. One of these variants does this quite fine, whereas the other one is clearly affected. They are, however, both similarly affected in the latency between choice making. So basically you put the mouse in front of the T-maze and both of these have a tendency to just run in and to make some kind of fairly random choice. If we do micro-CT, we see that all of these mice, all 3 variants have microcephaly. So they have a smaller skull. And if we make an endocast of this skull, then these endocasts, which are not brains, they're endocasts, they're also smaller. So basically these mice have a smaller skull with a smaller brain volume. Now the last slide I'll show is probably the most interesting one, and I got emailed this one just yesterday evening at 9 o'clock. So this is doing MRI. So this is where we're looking at the brains of these mice, and we can measure volumes and thicknesses of specific regions. So what we're looking at here is the wild type. So this is an average of 15 wild type mice and 15 mutant mice, where we're looking at cortical thickness. So this is taking the neocortex as a whole, so the bit that sort of controls our emotions and actions. If we look at where the wild type has a thinner neocortex than the mutant, you see that there's almost nowhere. Yes? Okay. Yep. Whereas if you look at where the neocortex is thinner for the mutant than for the wild type, then with very significant p-values, you can see different regions on the neocortex that are thinner than with the wild type. So this at least suggests that there is a contribution of development of the brain, but it doesn't mean that there aren't any problems with synaptic pruning, synaptogenesis, or with synaptic transmission. Basically, it doesn't exclude any of these hypotheses. So now that we have these mice, we are using genetic crosses to try to rescue these phenotypes. We are working on doing AAVs. There's of course the option of pharmacological metabolic rescue with OGA inhibitors or with sugars. Electrophysiology to see whether there is a problem with synaptic transmission, proteomics, RNA-seq. And our ultimate goal is, if it turns out to be a disease of hypo-O-GlcNAcylation, to drill down to individual O-GlcNAc sites within the proteome. So that brings me to the end. We have recently moved to Aarhus University where we have settled, and that's going really well. This is the team of people that work on O-GlcNAc and OGT-CDG. I'm funded by the Willem Foundation and the Novo Nordisk Foundation. A lot of collaborators in Aarhus that have contributed to the data that I've shown. We have several positions open if there's young people listening in that are keen to come and help us out. And I'd like to acknowledge that there's 5 young people that have come along. I'm an aging man, so these are the 5 young people that are actually in the lab doing the work. And so as parents, it's them that I think you would need to talk to and inspire, because it's their hands that is going to drive this field forward. And thank you very much for your attention.
We're actually behind the schedule, but I see that couple of hands, and we can probably agree that we are eating in our break. But I guess it's important to hear people. Question. Yeah.
Gulchen Pekurnas from University of California, San Diego. That was really great because I think mouse models are going to be really important to really understand the mechanism. And I'm wondering if you actually evaluated the brain sections, did any histology, because both the patient phenotype and the mouse behavioral phenotype indicate specific defects in inhibitor interneurons. And considering their developmental trajectory, I think there might be something there differentially affected.
Yeah, so we are doing this at the moment. Data aren't complete enough to present here, but actually there is one of the students that has come along with me has a poster on this that we might show at the the last session of the day where we meet for coffee, and then please do interact with them. Yeah.
Don,
Yep.
So John Hanover for those on Zoom. It's a related question. This OGT is found in really high concentrations in the 6th and 7th cortical layers for reasons that none of us have ever understood. Is the thinning consistent with loss of some of those cortical layers?
Yeah, so in the preliminary experiments we have done, we don't see this. But again, I would encourage you to go and look at Shahana's poster where you can see exactly that data. So we don't quite understand how cortical thinning could happen without loss of a particular cortical layer, yet that is not what we see. So this is interesting. Yeah, Irina.
So this is a question from parent.
Yes.
So there was a slide second from the end when there was a mouse, and you showed like you try 5 different treatment ideas with the mouse model. What are you measuring in those as an outcome? Are you looking at just behavioral changes, or you have ideas on biomarker?
Yeah, so the first stage of using a mouse model to look at treatments is to first establish very clear quantifiable phenotypes. So we need phenotypes you can measure with a hard number, because later on we're going to treat these mice and we're going to ask, is there an improvement? The improvement will never be 100% whatever treatment we try. But we need something we can measure very accurately so that even if there's a 20% improvement, which might actually be relevant for the patients, we can measure that and we can say that. So at this stage, we are building sort of a battery of tests that once we are satisfied with them, we're then going to apply them using the treatments that I discussed. And it's slow, and I'm sorry about that.
We've got a question from Anna on Zoom. Anna, please go ahead and unmute yourself and go on camera, please.
Yeah, I'm trying to go on camera.
Whatever works for you.
Okay, I'm here. So I'm Anna Dritikova, and I'm a molecular biologist by training. And the last 14 years I spent working in the field of AAV-mediated gene therapy, and have designed multiple vectors that are currently being evaluated in more than 10 different clinical trials in humans for various diseases. I'm not a subject matter expert, I'm not a CNS expert, but I do know how to make a vector that works. So my question for you is, in the mouse model that you created, have you seen any differences between the males and females? So did you look at the male carriers for the behavioral phenotypes, or have you created the homozygous female mice to see what happens in those animals in terms of behavioral readouts?
Yeah, so we only work with male mice, and the reason for that is twofold. Both from a breeding strategy, it's easier to make these. Also, at least in our list of variants, it is mostly males that are affected. Doesn't mean that the disease isn't important in females. The other reason is that I'm no geneticist, and I think this is maybe a question that either Stephanie or John can answer better. But I do know that axon activation is different in mice than in humans, and therefore it is questionable whether you want to make female variants if it's clear that axon activation plays a role in a mouse model, because it might not necessarily tell you much about what goes on in human. I see John Hanover nodding, so it means I say something that makes sense. That's great.
Thank you so much.
Yeah. Do you want to read one more question from Joe? If that maybe Daan, you can take that one. This was for a previous session, but maybe you could take that one. Are there any different isoforms of or splice variants to OGT? And if there are, could any be expressed differently in different cell types, i.e., neurons?
Yeah, this is absolutely possible. So there's a lot of work that suggests that there might be different isoforms of OGT that have fewer or more of these TPR repeats that Jerry Hart described in his first talk. It's not something that we have looked for at the moment, because at this stage we don't know whether, as is shown here, whether it's the expression of OGT or specific OGT isoforms that is affected, or whether it is rather hypo-O-GlcNAcylation, in which case that doesn't really matter all that much.
Thank you, Dan. Any more questions? Awesome. Appreciate it. Thank you, Dan. Right, the next step, we've got a combined presentation from Hudson Fries and Andrey Edmondson. Let me introduce Hudson Fries first, who will go with his presentation. Hudson unfortunately couldn't make it in person here due to some family reasons. I hope everything is fine. Hudson Fries, Director, Sanford Children's Health Research Center. Hudson, please feel free to connect and screen share your presentation. And after that, we'll invite Andrew on the stage as well.
It says the sharing is not turned on for me.
Can you try again, please?
Okay, will do. Oh. Do you see that?
Yes.
Okay. First, let me sort of give you a bit of a background here. Let's first ask about whether OGT is a CDG or not. So what we've done in the most recent version of defining CDGs is to say that anything that is about a sugar going onto a protein, either before or after it's put onto the protein, qualifies it as a CDG if you can find patients that have a phenotype, and then if you can take a model system and show that there is at least some of the features also work in that. That can be cells, it can be mice or whatever. So by that one, we are looking at a CDG. So, people can debate that, but that's how we're looking at it now. I think the important thing to point out here is that you are off to a great start. And the reason I say that is because you've got all the stakeholders here. I grew up as a glycobiologist, biochemist, but as you all have seen in this situation, the families are important, the physicians are important. And so we all have to be able to really communicate with each other. And I think by having this kind of meeting where you introduce really stellar questions, is a great way to kick off this collaboration. And I would just say, keep the communication open. And of course, we all know there is a balance between cooperation and competition. There's not enough money. There's not enough people working on this. And the families and the physicians are always saying, come on, can't you give us more? So where do you go when your doc doesn't have the answer? You turn to scientists. And that's where we are now. But in my decades of experience here, it has been absolutely invaluable to bring in the families. And I'll give you one example. Yu Yamaguchi, who works on multiple hereditary exostosis, had at one point seen a paper where it said that a few of these kids, in fact, they were brothers, had autistic features. And so he, at one of our meetings, talked to a mom of one of the patients and said, well, does that register with you? And she said, oh yeah, all our kids are that way. He then went on to write a PNAS paper to get some funding for it and to show that, in fact, there were linkages of the autistic features in his mouse model. And you would have never known this really, because this was a rather obscure paper if you hadn't talked to the family. So I just want to emphasize how important that is of sharing the information. You're off to a great start. There is another group that of patient organizations, which is called the CDG Care, and they sort of envelop all of the various types of disorders, and Duncan Webster knows that group very well and is part of it. And just recently, they obtained a grant from CZI, and that's the Chan Zuckerberg Initiative, which has been put together to advocate for diseases and rare diseases and how you can get that out into the public. Again, I think you're off to a great start, but it might be worthwhile to recontact them because, again, I view this glycosylation as across the entire spectrum, and it includes the O-GlcNAc transferase. Another item I want to point out is that the FCDGC, which is the Frontiers of Congenital Disorders of Glycosylation Consortium, is just finishing up about a 5.5-year grant that came from NCATS, and that's the sort of translational arm of NIH, and NINDS. And that has been remarkably successful. They've been able to enlist, and Andy will probably tell you about this, over 300 patients. The goal was 100. They have over 300. And so the idea is you have to have natural history, as you would need in the mice. I think Daan pointed that out pretty well. You need the natural history to know what the course of the disease is. Fortunately, you have the data that he and Lance have come up with for looking at patients. You need a biomarker, and I'm going to leave that aside other than say, yes, I think it's important. And I think that probably the best models to look at, in addition to the mice, and I'm very impressed with the data that you've been able to generate with all the different mouse models, is probably going to be IPS differentiation into 3D organ cultures, mini brains, call them what you will, and combining that together with spatial omics. So in the future, I think that's where the greatest advantages are going to be. We also need our organismic models, such as the mice. And I just want to say one thing about the media that you choose. And Lance pointed out a good thing, that we have many cell lines that may have evolved and taken on different mutations from their ample supply of glucose. And what that means going forward, I think, is a good question. But I would encourage people to try to get early passage fibroblast to make iPS cells, because those are the most successful ones, and maybe Daan has already done that quite well. Again, have the kinds of conditions that are important for being able to be physiological, because if you're not there, the translation is going to be difficult. So now what I'm going to just move into on my slides is just impressions about sort of basic glycobiology and therapy in patients, and coming from what we have done already. So we've had pretty good success with monosaccharide treatments of different disorders. Now, we don't know that it will apply to OGT, but I think it's worthwhile to point out that mannose can be used to treat phosphomannosubstrate deficiency. And also we're getting data that shows that neutropenia and glycogen storage disorders can be treated with mannose. And no one would have ever thought of that. Fucose can be used to treat leukocyte adhesion deficiency type 2. And now, because we found that GLUT1, the major glucose transporter in most cells and in the brain, certainly across the blood-brain barrier, is also a fucose transporter. And so we have some initial data suggesting that fucose might aid in treating GLUT1 deficient patients, and they could be 1 in 25,000. Galactose is being tested in clinical trials for PGM1 deficiencies, and SLC35A2, which is the UDP-GAL transporter. MANNAC and acetylmanosamine has been used to treat GNE myopathy. And I don't think the final word is in on that, but again, you can see that there seems to be some beneficial effects. GLcNAc has been used and is in clinical trials now to treat multiple sclerosis. And that's thought to go through the immune system and have to do with N-glycosylation and the formation of multi-branched end-linked glycans. But it also seems to be able to treat allocrima, which is the lack of tears. One parent decided to give his child GLcNAc, and even though he didn't make tears before, after being on GLcNAc for a week, he made tears. So a clinical trial is now being initiated in that in N-gly1 deficient patients. And finally, there is evidence that an aldose reductase inhibitor, that would be taking glucose to sorbitol. I don't know many glycobiologists who think about the polyol pathway, but it's been shown both in some fibroblasts in model organisms and even in a couple of patients that it improves their symptoms. However, the clinical trials have not worked out as optimally as people would have expected, and yet there were patients who showed improvement. So my point is that monosaccharide therapy may be right for some disorders, and it may have to do with a particular kind of mutation, dimerization, catalytic activity, location within the OGT itself. So I'm sure everybody is already thinking about that. Let me just give you a reason that I want to emphasize here. We've been working on fucose for a while, and Lance knows this well, but you realize that fucose can come from, can you see my cursor at all? Yes?
Yes, we can see it.
Okay, so fucose can come from outside, where it gets activated to GDP fucose. Fucose can also come from salvage pathway. In other words, the degradation of glycans that then release fucose and goes into GDP fucose again. It can also come from the de novo pathway where mannose or glucose gets converted to GDP mannose, and then those are able to go to GDP mannose here or here. GDP mannose is then taken in to the Golgi where fucosylation occurs by a GDP fucose transporter. So what you realize is that there are a couple different ways to use fucose, and that fucose can be then distributed into different glycoproteins. Well, we would have thought that all of those pools are the same, that they're all mixed up together. But we have evidence, and I'll show you just a couple slides, that GDP fucose coming from salvage is recognized differently than fucose coming from the outside. It's also recognized differently as GDP fucose coming from glucose or mannose. And I don't think any of us would have ever predicted that. But if we look at HepG2 cells, for instance, and we look at the fucose that comes from exogenous fucose, and notice that this is a very low concentration, just a few micromolar of fucose in the presence of 5 millimolar glucose. And what you see is that the origins of fucose coming from glucose and also mannose are almost completely suppressed at this level. The level that we have from the turnover of glycans, that's from the salvage pathway, is impacted much less. And we can measure all of these different contributions because we use differentially labeled sources of fucose, and we preloaded the cells for 11 days with fucose exogenous to saturate this pathway. And we could then look at the different proportions, and what you can see here is that if you plot this as a percent of maximum, you can see that certainly almost all of this goes up to 100% by that point, and it has a fairly minor effect on salvage, even though they're using the same enzymes to do both of those reactions. Somehow the cell knows the difference. Also in the inhibition of GDP fucose derived from glucose is handled differently than that derived from mannose. So we do this by looking at glycoproteins that are secreted. So we have a good picture of those things that must at one point reach into the glycosylation pathway, be glycosylated, and then thrown outside the cell. So this is, we think, terribly important. So if you're thinking about different cells, different proteins, different sources of GLcNAc, that could be GLcNAc, it could be glucosamine, but each one could make a different contribution in a different cell line, and we don't know that. And right now, because we've seen similarities in the way fucose is handled in the brain of mice from GLUT1 deficient models, we have a belief that there are probably different proteins that are being influenced by that. So my advice at this point, which is not news to anybody, is to get an assay, and there are multiple ones being developed. I would suggest screening approved drugs. That has shown some real benefit already in some of the patients, some of the other types of CDG patients. Andy may be able to talk more about that. And the only cautions I would have is that, again, different cells may rely on different sources of GLcNAc or glucosamine. And if you're thinking about that, you need to be rather careful. And finally, the other thing is, for instance, OGT overexpression could be toxic in some cells. We saw this in GLUT1 deficiency syndrome, where if you overexpress the GLUT1 transporter in neurons, it kills them. So again, one needs to be careful how you do this, and each case may be individually different. So with that, I'll stop and take any questions you may have.
It doesn't seem that we have questions in the room, but I will invite Andrew Edmondson to the stage. I appreciate it. Thank you, Hart. Thank you for this presentation. Andrey Edmondson, Assistant Professor of Pediatrics, University of Pennsylvania, Children's Hospital of Philadelphia. Thank you. Thank you, Andrew.
Now on. All right. So thank you for inviting Hud and I to speak together. We both work together in the Frontiers and CDG Consortium, and I think as Hud indicated, we're happy to include OGT as a CDG. So I want to show you some of our resources, as well as bring some sort of practical discussion on this conference about how to move into, treating actual patients and some of the things we need to consider. So the Frontiers and CDG Consortium is an international consortium. We have sites across the United States and in Europe as we're working to develop treatments for CDG. And our framework really is around preparing for clinical trial readiness. And so as Hud mentioned, we have an arm focused on natural history of CDGs, on the biomarker discovery, and then ultimately on getting into clinical trials. And some of my work has been most closely in the consortium with trying to move to clinical trials. I'm a physician scientist. I run a CDG clinic at the Children's Hospital of Philadelphia. I do have a couple of OGT patients, and we've been working hard to move in the clinical trial space. So I wanted to just give some overview perspective of what needs to happen in order to get into clinical trials and what are the types of things we need to be thinking about developing so that we can do that. And as was indicated in the very first sort of introduction, you need an idea. That's the very first start of all of this, is getting an idea. And I think there's a lot of ideas here. But once we have the idea, what do we need to do to be able to move this to get into patients? And so the next step is you have to develop that idea. Developing the idea is a lot of these things that we're already talking about. How do we start testing those ideas? Finding out, are these things going to actually move to be able to change things for patients? And those go into these preclinical studies, and mouse models are a really important part of those preclinical studies to determine, is that idea and the development of the idea actually likely to lead to success? And then it goes into a regulatory review where, and this reflects largely the process in the United States, so there is that bias here. But reviewing all of the development and the preclinical studies to really suggest whether this can move into a clinical trial. So if it passes the regulatory review, then we're able to move into the clinical trial. After a clinical trial, there's more regulatory review. How did this idea actually function within human subjects? And then that, if it successfully passes, that leads to market release. And then even after a drug is approved, then there's this additional post-market surveillance. So when you have a new drug and you start with the idea and you development and you do the preclinical studies, most likely you have to go back to the drawing board a lot of times. It doesn't sort of pass the muster. It doesn't have the effect you expect it to have. And this happens over and over again and again. But finally, once you get to the regulatory review, in order to try to move to a clinical trial because things look good, you have to put together this thing called an investigational new drug application. This applies even for sort of approved drugs that you now want to have a new indication. You want to use that approved drug in a new way. And what are the types of things that the regulatory agencies are going to require? Is mostly up front, they want safety data. They want to know, sort of with the Hippocratic oath, we're going to do no harm in our pursuit to try to produce benefit. They want to know that there's likelihood that there's going to be benefit. They want some efficacy, some evidence that there's likely to be an improvement. And then they want to know a lot of plans about how this clinical trial is going to happen. So they want to know what's your purpose, what's the dosing, how are you going to, how much are you going to give of your drug, How frequently are you going to give it? How are you going to ensure that it's safe? How are you actually going to enroll patients and recruit them? What is going to be your outcome measure? And they want this pre-specified. What do you expect to improve? How are you going to measure that improvement? How are you going to determine that the improvement, if you see it, was actually meaningful and actually has some meaningful improvement for the patients? They're going to want to know your statistical plan and how you justify that, and they want to know your quality assurance and that ongoing monitoring. So they're going to review all of this, and they need all of these things upfront, pre-specified, before they're going to allow you to move into giving this to patients and doing a clinical trial. What's the fastest path for this? So there's a lot of that upfront work that needs to be done. And so what a lot of rare disease groups are doing, and what we've been doing in CDG as well, is actually to try and take approved drugs, things that made it through some regulatory review before, use that as our idea where we don't have to go back to the initial drawing board and try to get that into a regulatory review where there's already a lot of that safety data that's been generated. And as long as we can generate some efficacy data that we think there's a reason that this approved drug should work in the condition, we may be able to move it more quickly into clinical trials. So that's some of what we've been doing in the CDG consortium. And then this is just an overview, and I'll skip through this quickly just to make sure we have time for discussion and questions of clinical trials. And so there's different phases of a clinical trial. There's different purposes to those phases of a clinical trial. And so some of the discussion of where you want to be with OGT-XLID ID in a clinical trial, and how you'll position that, and whether you want to be trying to get something into a single patient and primarily looking at safety, or whether you want to get this into a larger trial where you're testing in multiple patients and leading to regulatory approval. So with that, just want to give you the little bit of the overview and then open up for questions to try to get some practical thoughts about where you are and where you want to be.
Let's take a moment. Do we have questions? Yes.
Judy Fridovich, Kyle from Emory. So if I understand it, at this point, patients are identified clinically. They've shown symptoms, and after undergoing basically a diagnostic odyssey, they finally get a diagnosis. Is that correct?
Yeah, I think the vast majority are a genetic diagnosis at this point. A child who has something like we heard from the parents, that they were starting to miss their milestones around 6 to 9 months. They get genetic sequencing, and then they identify a mutation.
So by definition, any intervention is going to be trying to either halt or slow progression. Do we believe that this is a progressive disease or no? Or is going to be trying to reverse damage? And so I'm sitting here, having studied a disease for 33 years that has newborn screening, and that sounds to me like what you guys really need. And so I'm wondering if anybody is looking at dried blood spots from patients and from controls to say that's where we need a biomarker,
Yes, so that's a great question. If you're here tomorrow and in the session in the morning, sitting next to you is Ernest, who has actually been doing dry blood spot analysis for glycosylation changes, not specifically OGT, but certainly something in this space that we're sort of interested in. And I think that's one of the questions, though, with this condition and with many CDGs, is what amount of the phenotype is from some sort of a neurodevelopmental process that maybe happened before the diagnosis, how much of those disease symptoms are due to ongoing lack of glycosylation or problems and impact in the pathway, and which aspects can then be intervened to change. And that goes back to the thought of, if this is a sort of an architectural change in the brain, you can't really go back and change that. But if this is something with transmission in the brain, then perhaps you can normalize that and improve a fair amount of the symptoms.
Yeah,
absolutely.
Oh, I'm sorry. I was just saying that if babies are identified after birth, even if some of the damage occurred before birth, so you couldn't prevent that, oftentimes you can actually offer benefit even if you can't make it perfect after birth.
So, Gulcin Pekurnas, in addition to CDGs, the genetic cause of intellectual disabilities are not very well known, and I'm sure there is some other genome data coming from patients are out there. I'm wondering how much of collaboration is happening between different clinics. For example, just going into the data set for intellectual disability, maybe OGT mutations will be identified there too. How much of this collaboration is happening, and is there a way to kind of facilitate that so that more patients could be diagnosed and recruited?
Yeah, I think it's a good question. Certainly in the diagnostic space in the United States, we as a consortium are really trying to reach out to all of the major genetic testing companies who identify these variants so that then they can facilitate getting families connected to these studies. It's challenging just with the regulatory sort of environment of the United States in that private companies aren't allowed to force or publish the data of all of these patients. So we're restricted in basically asking companies to provide information to the physicians who ordered the test to then pass it on to the patients, and then it's reliant on the patients to reach out and connect to us. So we're trying to facilitate that, and so for engaged families who reach out to us, we definitely have those. But how many families are diagnosed and given the information and are just so overwhelmed at the time that they don't have the bandwidth really to reach out and engage in research, I think is a challenge.
I just want to add a comment about the discussion to newborn screening. So APGAR scores of these newborns are completely normal. So the developmental delay is only identified after about 1.5 to 2 years. And this is an X-linked recessive inheritance syndrome with mostly de novo variants. So there is nothing to screen for because we don't know which variants in OGT are causative or not, or so it would be impossible to make a gene panel that you could screen for a particular mutation because they're not spread in the population. So this idea that was just muted won't work.
I would say the caveat to whether or not it will work is back to the biomarker discussion. And if you're not doing a genetic test, but you're doing a metabolic change. So most of what we do in our blood spots is actually metabolic changes, and then you follow those up with further genetic testing. So if you can identify a biomarker that ideally precedes any clinical symptoms, you have a much better window to intervene.
It doesn't have to be a metabolite. I mean, galactosemia, that test is an enzyme assay, crude, but it's an enzyme assay. So if you could test for this enzyme in a dried blood spot in a high throughput way, you could have a newborn screen.
Yeah, so there's a variety of approaches that could potentially be used as biomarkers for this and other conditions.
I'd just like to add that, the thing that Jerry brought up in the beginning, that there's almost no variance in OGT. And so if you see a variant, it's much more likely that it's something that will correlate. I mean, we had a situation with a gene called CAD, 2,250 amino acids, and there are 1,000 variants, coding variants. So, it was hard to tell which one, would be damaging. And of course, you have to have two separate alleles to do that. And, fortunately, we were able to design an assay for it. But, with OGT, you may be somewhat luckier based on the absence of many variants in the population.
Thank you, Hudson. Thanks, Andrew. With this, we have a 10 minutes break. It was 15, but can we just shrink it to 10 if that's okay with you? So we'll be back at 55, that's okay? Right, guys. Let's get started. Our next speaker is Natasha Zachara, Associate Professor of Biological Chemistry, Johns Hopkins University of Medicine, and Stephanie Olivier-Van Stichelen, Assistant Professor in Biochemistry in OB/GYN Medical College of Wisconsin. And we're going to hear about the OGA inhibitors, biomarkers, and what we could do there. So please welcome Natasha and Stephanie.
So I'm Natasha.
I'm Stephanie.
And we're not really going to do a presentation today. What Lance pitched to us, yes, Lance, that was you, is that he wanted to have an open discussion about the potential for cures, but also for biomarkers. So what Stephanie and I've done is prepared a few slides that kind of lay the foundation. But then we want to have this discussion about what everyone knows in the field, and we'll take notes, and then we'll eventually send that out to the group.
Yeah, exactly. Okay, I'm gonna take the first bit of this. Okay, so we're gonna talk about two different things, which I think are the hardest part of most of that today, which is the biomarkers issue that we've been talking about, and then the various tools that we already have, that some of them already make a little bit of the clinical trial, such as some inhibitors, some that are freely available, like metabolic substrate. And we're mentioning gene therapy here as a tool. However, we're not going to personally talk about it because there's the talk just after us about that. Okay, so where can we intervene on getting somehow this O-GlcNAc cycling back on track when you have an OGT mutation? So it was not mentioned too much today. I feel like that O-GlcNAc is added onto protein by taking this UDP-GlcNAc molecule. And so this UDP-GlcNAc is made out of glucose, and this is the exosamine biosynthetic pathway that's basically a series of reactions here to make that UDP-GlcNAc. A couple of things we can do eventually would be to use those substrate here, glucosamine or N-acetylglucosamine, to kind of feed in that pathway and give more UDP-GlcNAc for those deficient OGT proteins. Another thing would be to target directly OGT, and that would be more gene therapy. And then another way we have already available OGA inhibitors that some of them, again, are in clinical trial for Alzheimer's disease that have made through a part of that regulatory phase. So that could be more readily available for us to use. And that would actively basically block the OGA and maintaining O-GlcNAc onto those proteins. So what do we have? So those glucosamine and GlcNAc basically is freely available, and you can buy this on Amazon, I believe, or yeah. So I did not know this, that it was still freely available. But after talking with a couple of parents, they told me that they're actually already using GlcNAc somehow in their daily life. So I think this would be nice for you to give us like your feeling on this as well. But basically, like I was saying, those two, glucosamine and GlcNAc, again, supplement just freely available, would fit in that pathway and kind of like increase this UDP-GlcNAc. And hopefully gets more GlcNAc onto proteins. The second part is this series of OGA inhibitors. Not all of them are in clinical trial, but some of them are. They do have different design, either mimicking different part of that process, but overall, the goal is to inhibit that enzymes that remove O-GlcNAc. And then again, gene therapy, we're not going to talk about it because we have a talk just right after us. But basically, the idea would be to rectify those mutations on OGT. Okay, so a couple of outstanding questions we've identified. And again, this is really meant to be a discussion here. So we did throw some stuff in there, and we have thought about it with Natasha, but we do want your input now, and we'll take notes. First is, what should we consider right now using those substrate therapy, either GlcNAc or glucosamine, or even going into the OGA inhibitors, particularly for OGT-XLID patient? Should we do one question by one question, and then maybe we can pitch in, or?
I think we should read all three of them
Okay.
because we have some answers on the next few slides to kind of anchor the discussion.
Yep. Okay, so, and then a theme that came up today again is that all those OGT mutations are basically family specific. So should this consideration using GLcNAc or a substrate therapy or OGT inhibitor different based on the OGT mutation the patients are carrying? And then what are the possible benefit and risk of the different methods that we've just presented?
So this is where we want your help. So Stephanie and I started thinking about this already. And so we see different pros and cons, as well as some different considerations for each of these different approaches. So we know that metabolite supplementation is well tolerated, and it's in clinical trials for treating, at least for GLcNAc, IBD, and multiple sclerosis. And they've actually measured the effectiveness of dosing patients with 2.5 grams of GLcNAc per day. We know that OGA inhibitors are in clinical trials and will be more specific than metabolites, for instance. And we know that gene therapy offers the appeal of correcting the disease and having almost a single therapy for all of the patients, which is, again, appealing.
I know you were on the roll right now. Disadvantages that we've identified is, like it was said, I think by earlier, metabolic supplementation can impact other glycans. So what off-target effect are we going to have? Metabolite or GA inhibition supplementation may not be effective for all mutations identified in OGT-XLID. And OGT inhibition or gene therapy, what are the effect on OGT or OGA? What is this one? Wait.
This refers to this kind of homeostatic response of O-GlcNAc, that when we delete the O-GlcNAc transferase, the O-GlcNAcase protein abundance goes away in an attempt to correct the levels of O-GlcNAc. So there is this danger that if we use virus to overexpress OGT in mice or in cells or in patients, that one consequence could be that we lower the levels of the O-GlcNAcase, causing a completely different issue. Now, I think, in fact, in this case, that this is a situation where this may be more different for catalytic domain mutants versus TPR domain mutants, because I think we'll get less of this effect when we have catalytic domain mutants of OGT. So then we also have some consideration. We don't know how effective metabolic supplementation is in the brain. We haven't measured it. I mean, we have to measure UDP GLcNAc levels in mice that have been treated, but being able to do that and being able to get out to harvest the tissue without getting a lot of quenching of the metabolites is going to be really challenging. We think that maybe OGA inhibition, which can alter the abundance of OGT, could have some impacts on non-catalytic and scaffolding roles of OGT. And this is a big one that's come up several times today. Do we have a biomarker for assessing treatments? And that's going to be really important as we approach this problem. And then, and I think Dan brought this up a little bit today, and I know this, I have a colleague in the IBD field who's using GLcNAc to correct some of the symptoms of Crohn's disease, and they have a questionnaire that they give the patients. So there they put them on these crossover trials where you are your own control. So you're blinded, but you might take a drug treatment, so you won't know if you're getting GLcNAc or you're getting glucose. And you take that and you fill out the questionnaire, and then you have a washout period, and then you take the other drug, right? And so it's a way where you're your own control, but they have these very detailed questionnaires that the patients answer in addition to taking serum samples and other samples, so that you have a qualitative patient of life assessment as well as a more molecular assessment.
I guess, can we maybe open it so to additional like advantages consideration, and we'll just fill the spreadsheet we have in front of us.
So Andrew Edmondson, I was just speaking in the last session. One thing we didn't bring up that maybe is a little bit relevant to this goes back to your metabolite supplementation in that we actually are just starting a clinical trial using GLcNAc in GLY1 CDG. So I know some of those aspects, and I would say the safety aspect and stuff is pretty easy to move forward. The big question is it actually going to be efficacious, and how are you going to measure that? Or like, what do you think is going to improve? So the path that we've used in GLY1 was a N of 1 safety trial in a patient and tried to determine what are the symptoms that improve so that we could then justify measuring improvement in that symptom in a larger clinical trial.
Another question maybe for you also is like the N of 1, particularly in like rare disease like this, how is that like affecting, I guess, like the regulatory part of this? Like how many patients do you need?
So N of 1, you need one patient by definition, and it's done to.
But like I'm wondering like how you measure this and how like we think N of 3 all the time.
Yeah, so for N of 1, you're really looking at safety, and then you're just asking patients to sort of say what they see improved as well to try to get information on how you're going to be able to power and do a larger study. So really the number you need for a larger study depends on how big of an improvement you have and how you're measuring that. And what's going to be statistically what is required in order to have enough patients. Typically in our rare disease space, we get into the order of 20 to 40 patients for the types of improvements we hope to see in these metabolite supplementations.
Thank you, Judy Friedovich, Kyle, Emory University. So I think one of the challenges, so I study classic galactosemia and other kinds of galactosemia, and one of the challenges we face and that I suspect you may also face is that there is incomplete penetrance and variable expressivity for just about every outcome domain that people with galactosemia, where they tend to have more complications than people without galactosemia. And so when you do a clinical trial, I think you have to be very mindful that you want to make sure that you are including people who are likely to have a problem and not likely to do well anyway, because of the risk-benefit ratio, and sort of your N is likely going to be determined by what, if you are starting pre-symptomatically, what fraction would you have expected to show, problem A or problem B, so that then you have the statistical power to say, we made a difference or we didn't. So I'll let you respond to that, and then I have one other.
I totally agree with you, and I think that's why we raise this question about some treatments being kind of background specific. But I actually think often the only opportunity we're going to have to treat patients pre-symptomatically as if they have a family link to other patients, and that is one of the things that's going to make any of this just a little bit more challenging.
Yeah, so my other thought was about the biomarker. So there are biomarkers that are associated with the condition, and you can use them for diagnosis, but you don't know what's causal. So if you are looking for let's say it's a small molecule. If you're looking for a small molecule that you want to use for diagnosis, all you need is a really strong association. But if you're going to try to use that biomarker to follow treatment and, effectiveness of treatment, unless you know that biomarker is actually causal, I think you may end up having problems.
Yeah, I agree. And again, I think that the biomarkers for the catalytic domain mutants are going to be a little easier to interpret than the TPR domain mutants, just because we see a much bigger change in glycosylation on those. Lance.
So what you're saying is basically we almost have two moving targets at the same time. Like we're like looking for like treatment options, and at the same time biomarkers, and both needs to kind of like match.
But biomarkers for diagnosis may not all be the same as the biomarkers for assessing treatment options, because biomarkers for diagnosis may not be causal for the disease. It just convenient.
Yeah, and kind of following up on Judy's point, I mean, I think we have to build, especially with metabolic supplementation, OGA inhibitors, we have to build trials that are likely to work. And what that means probably is having in vitro data that they have a kinetic problem, right? If you have a KM variant to sugar nucleotide, increasing sugar nucleotide should help. But if you have a TPR variant where the KM for the UDP-GLcNAc is exactly the same as wild type, I think it's highly unlikely that supplementation is going to work. And so we need to build trials that are going to work,
But
right? Or you're going to get stuck, right?
I think this is also where the parents can help us, right? Because if patients with TPR domain mutants, you are seeing an impact with the metabolite, then it's telling us something that we did not anticipate.
Correct.
I actually want to continue right after Lance's point, because so if we really understand the cellular mechanisms and what is happening, instead of only focusing on supplementation to alter glycosylation, we can also focus on the other metabolite treatments. For example, we know from our studies that if there are alterations in OGT activity, that significantly affects neuronal metabolism, and there are ways to boost up neuronal metabolism. For example, you can add additional mitochondrial supplements, additional vitamins, and these treatments are already being used for mitochondrial diseases. So better characterization, target-specific characterization, I think is going to really create almost metabolite cocktail treatments.
Yeah.
There was Andrew and then
I'll just follow up related to biomarkers, but taking a step back from that, when it comes to clinical trials and moving these into patients, is outcome measures. So oftentimes we think of biomarkers as an outcome measure, but in order for FDA to really approve a biomarker as an outcome measure, it needs to be associated with the disease and reflect what's expected to be disease symptom output. So it needs to be sort of directly related. It can't just be a random metabolite that you're saying is improving if it's not associated with some aspect of the disease pathophysiology. But you need to connect all of those points when using a biomarker as an outcome. But you don't need a biomarker, like a measurable thing that has to be an outcome measure for a clinical trial. So you can have other, functional improvements in your questionnaire is another one of those, of like patient-reported outcomes that they also do like to have, and they want to be able to measure those to make sure that, particularly if you have a measured biomarker as part of the study, they want to make sure this is actually causing an improvement for patients as well. So just because a biomarker improves doesn't mean there's any impact on the patients, and we want to make sure that the patients also find this to be a reasonable improvement for their kids.
I'd like to.
The biomarker will also give you a way of reporting symptoms that you possibly aren't so enthusiastic about as well.
I'd like to follow up on one of the comments there. First, I agree with Andy that you may have a biomarker for diagnostics, and that may not be effective for any kind of treatment. We've seen that in several CDGs. And I think that's really going to be important. The other thing is because GLcNAc and glucosamine are available online, parents have certainly looked at nanos and triacetyluridine and things like this that they can get online without the benefit of going through a clinical trial. And of course, we can't keep parents from doing that. But the point is, if you have good communication between the scientists, the families, and their physicians, if everybody's together on the same page, then at least you can agree on, all right, you may go off and do that, we can't prevent that, but please provide this kind of information if you're going to do that. More coordination is going to be really important for those things that are easily available.
Can I follow up on that comment, please? Because actually that is related to something I was going to bring up here. Is that all right?
Sure, go ahead.
Okay. Yes, so I mean, So I guess to start with that, but then I want to jump back a little bit. So one of the things that you will really need to get these trials planned appropriately is a really good idea of the natural history of the disease. And so, engaging with your parent communities to do natural history studies. And then it sounds like you have, despite all this genetic heterogeneity, it seems like you may have a pretty well-defined and reproducible clinical phenotype, which is good. One of the things that's a little scary is that if you have all of these supplements that parents may or may not be taking or giving their kids that may impact the disease in some way or may make things a little bit messier in terms of the natural history, that will be harder to establish what your trial is going to look like later on, because your data in your natural history study will be a little bit more of a mess. And what you really want is things to be as sharp as possible to get companies interested in pursuing this. And so, like Hudson was saying, people are going to do what they're going to do, but you really need to have some level of control over having people disclose what their patients are on, so that you can understand whether or not any kind of study you're doing with patient progression is or is not confounded by that. I guess the one other comment related to this kind of in general is that people are doing a lot of talk about using repurposed drugs and things like that. And there's a lot of variables being discussed right now in terms of what's going on with the disease and then what are the possible treatments. This is probably also a good time to see if you can identify industry collaborators in those areas that are running the trials with those drugs and things like that, because you'll decrease your number of potential directions by identifying people that are definitely not interested in lending you their drug. And you may ideally identify people that are interested in moving forward with helping you sort things out as you get the information you want. That'll at least decrease your number of directions you are talking about going in at a time where you've got a lot you can do.
Okay, I think we have one last comment from a parent, and then we need to move on to the second phase of this discussion.
Yeah, I just wanted to share some insights from the parents who give us supplements for quite a while. So I know a family who give glucosamine to a kid for 8 years by now, and he's 16 now, non-verbal, not potty trained, very delayed. And I know another 16-year-old girl who has never got any supplement, and she goes to grade 9 in school, though she needs to be in grade 10. So it's like all over, right? We give to our daughter, we started to give her glucosamine a year ago. GLcNAc, we started with GLcNAc, and we have been giving it for 9 months. There were no breakthroughs. So the problem is the kids are developing, right? She's still developing very slowly, but developing. So I don't know, is it due to supplement, or is it due to a therapist per week that she's getting, or maybe, this one-to-one that she's getting? So my question, Andy, to you, when you have this N01 trial and you base your outcomes on the questions from the parents, do you do it on the kids who grow or adults when you can have a kind of fixed? Yeah, so do you do it for the kids? Because when you give something to the kid, you develop by itself, right? How do you identify, is it due to the supplement or due to other things?
Yeah, it's challenging. For an N of 1, you're really looking at safety, not efficacy, and we're just asking families to share with us what they see is improving. And I think that is a perfect example of the challenge of N of 1 and of these supplements, that if it's doesn't look like it's helping someone and maybe it was helping someone else, or you have this variability of someone who wasn't even treated and they're doing so much better than one who was treated. Is that part of the supplement? And so they're not doing so well because they were supplemented. There's a lot of questions, and that's where a clinical trial with a blinded phase helps you to tease those apart. But it's a challenge. It's another challenge we've noticed with these supplements and families. If you really think the supplement is doing something important and good for the child and it's available, and now you're trying to do a more definitive clinical trial, how do you convince families to go off of this treatment that they have access to? So it's a very challenging space.
Okay, so we really need to move on to talking about biomarkers, which I suspect will be even more controversial and have even more ideas than this marker. And in the interests of full disclosure, I drew the short straw because neither Stephanie or I work on biomarker development. So I borrowed this and updated it from the NINDS website. So as we heard a little bit about before in the talk before, biomarkers take a ton of work to develop. There actually aren't really a lot of biomarkers that are used. There's a discovery phase, an analytical validation phase, there's a clinical validation phase, at which point you can return to zero and have to start all over again. You then have a qualification phase that we've already heard about from the FDA before these things even go into clinical practice. And so Stephanie and I started thinking about this, and we had some questions, right? You would really ideally want a test that could be performed on proteins from blood cells or punch biopsies or urine, something that's easy to collect and not super invasive. Should we be looking at protein, or should we be looking at something else like RNA or DNA? And which approach? Sequencing DNA, mass spectrometry, ELISA, what is going to be an effective type of test that is easy to establish and use in different laboratories? Because I can tell you, we can't agree on how to run an O-GlcNAc Western blot in this room. So the other question, so I think we had some more questions. What should the target be? OGT, OGA, O-glycosylated proteins or other molecules? How should we design specific experiments to identify novel biomarkers related to O-GlcNAc signaling? And then how do we validate markers and make sure that everyone in this room can detect the same thing on the same set of samples? So we, again, came up with some considerations, current tools and challenges to anchor this discussion. Ideally, a test that can be performed on something that's simple and non-invasive, like blood or urine. I guess this is a little bit of a repeat, actually. Protein, RNA, DNA, sequencing, mass spectrometry, ELISA, standardizations of approaches used in discovery experiments. So every person who gets the same sample gets the same result, if that's what it's supposed to be, so that Dan's mouse looks like Lance's mouse, or Dan's ES cells look like Lance's ES cells when we look at that data. What are the current tools that we've got? We've got a whole lot of pan-O-GlcNAc antibodies and readers. We have some site-specific antibodies. We have some techniques such as proximity PCR for assessing GlcNAc on a range of proteins. There's the possibility of an approach called parallel reaction monitoring that uses a mass spectrometer. We can assess OGT and OGA activity, and Dan alluded to a new approach today that I'm going to be excited to see, whoever's poster that is. Changes in OGT and OGA splicing, that might be something that's relatively easy to address. And looking at OGT and OGA abundance, possibly by ELISA, which is something we really don't do a lot of right now. I think the challenges are going to be identifying the targets, because depending on which organ you're looking in, we're looking at really different proteomes in those. And so identifying something in blood might not be good in urine, might not be actually something you're really even looking for in the organ that's most affected by the disease. We need targets that are non-responsive to inflammation and diet, because we already have a huge amount of biological variation, because each of these groups of mutations come from different families. Common or specific targets for, I don't know where I was going there. I think that might have to do with common or specific targets depending on which mutation. Like, are we going to have one set of targets for TPR domain mutations and one set of targets for catalytic domain mutations? And how do we go about validating and cross-validating these targets in different models? And so now we'd like to hear your thoughts.
So in the interest of time, because we're running late, We're going to come back to biomarkers a lot in the workshop and beat that to death because I think most of us think it's the most important thing to find right now. So I would suggest we just move on, if that's okay.
Sure.
Steve. Thank you, Stephanie. Thank you, Natasha. Right, next up, Liujiang Song. Liujiang Song is a director of the preclinical department of M34, and Liujiang has been doing AV gene therapies for and researching that for over a decade now. And we've asked Liujiang to talk about the gene therapies for OGT-XLID, but specifically maybe focus on AAV as a potential avenue. Please meet Liujiang. Liujiang is joining on Zoom. We cannot hear you. No, we cannot hear you.
How about now? Hello? Hello? Can you hear me, please?
We can hear you on Zoom.
Okay. Can you see my screen?
No, we can see you though.
Up to a second ago, we could see her screen on Zoom too.
Oh, okay. I see the comment. We can hear her on Zoom. Okay, that means that's probably not on your end, Liujiang. So let us check out here.
How about now? Can you hear me? Can you see my screen, please?
On our end.
We can hear and see on Zoom.
So you should be fine. It's on our end. Yeah, you should be fine. Give us a moment. We will try to resolve that in the room.
Thank you.
Thanks, Joe, for flagging. Yeah, thanks, Anna. Can anyone else go just on Zoom, just unmute yourself and just try to speak? Hudson or
trying now.
Yeah. Can you hear us?
We cannot hear anyone.
On Zoom, we could hear you.
Okay, now all good, guys. We can hear you. Right. The jump.
Hi. So you can hear me and you can see my screen, right?
Perfect. We can see your screen. We can hear you. Apologies for this technical issue. Thank you.
Oh, sorry for the bugs. So thank you, and thank you, Andrey, for coordinating this meeting and for the kind introduction. As you've mentioned, I have been working on AAV for more than a decade. So AAV is what I love. I love the research. So from now, I'll switch the gears a little bit for this meeting. I will take the next 15 minutes to talk about some basics of adeno-associated virus, AAV, and its application as a gene delivery vector in gene therapy. But before I go any further, I'd like to show you this quick video that I borrowed from my mentor, Dr. Jude Samuski. So I guess what you see here right now is a layer on the screen. But as we go down the magnification, here you should be able to see a human hair on the head of the layer, right? If there is no delay on your end. So if we keep going, now you should be able to see the spot in the middle of the picture. And if we zoom in, you will see more things come up. And here you will see the red blood cells, white blood cells, just to have a sense of the size of these different kind of organism and cells. So as if we come up with at this magnification, this yellow ball-shaped thing is the streptococcus that sometimes cause my son's strep throat. And this is the E. coli, the green thing. it's the E. coli that gets you sick when you don't cook your hamburger thoroughly. So as we zoom in further at this, you should be able to see this awkward looking structure. This is actually Ebola virus and how it appears under the electron microscopy. And as you continue to zoom in to about 20-25 nanometers, this is the elegant, beautiful particle that we call adenyl-associated virus, AAV. So AAV actually is a member of the family parvovirus. Parvovirus are physically small and genetically compact viruses. They measure about 25 nanometers. The whole AAV particle consists of two components: the capsid protein shell, and the shell actually decides what types of cell that AAV could infect. And also inside of the shell there is a DNA genome which carries all the genetic information into the cells. AAV has been considered as a non-pathogenic viruses, and it was initially actually isolated at the NIH in 1950s during their efforts to identify what was causing the common cold and to identify virus for vaccines to protect individuals. And it was first reported in 1965 as a contaminant of adenoviruses. As you can see here, there is a little particle around 20 nanometer size. This is AAV. This in contrast to the adenovirus, which is about 100 nanometer. So after its discovery, there was not much research interest in this virus because it doesn't really cause any known disease. However, Dr. Ken Burns, he was fascinated by this small virus, and he brought this virus from NIH down to the University of Florida, where a group of pioneers, including Dr. Nick Muzichka, Dr. Arun Srivastava, Dr. Judy Smuski, they started to study this virus more in detail, and their pioneering work made it possible to transform the AAV from almost a virus to an amazing vector. So in 1996, AAV was first used in human clinical trial, and let me go fast forward a little bit. So in 2012, this was the year that the first AAV drug got approved in Europe here. And in 2017, Luxturna became the first AAV drug approved by the United States FDA. As of today, there are 8 AAV drugs in total, including Zolgensma for SMA, which has treated maybe more than 3,000 children, and there is AAV drugs approved for both hemophilia A and B and also Duchenne muscular dystrophy. Besides this, there are about 290 AAV clinical trials either completed or ongoing currently, and the FDA anticipated an increase in accelerated approvals for gene therapies. This is obviously a sign that there is growing confidence in the therapeutic potential of AAV to treat maybe wider range of disease, which I think is really exciting. However, when we talk about AAV as an incredible vector, I believe it's also important to tell people or to recognize that there are also challenges and hurdles. So such as manufacturing, and currently all the approved drugs basically cost millions of dollars per treatment. And part of the reason is because of the high cost of the manufacturing. So obviously the high cost potentially could prevent many patients from accessing the treatment. And another hurdle is that AAV actually is such a common virus, and many of us may have been like previously infected by a wild type strain. So we know that when we get infected by AAV, it actually typically doesn't cause any symptoms or problems. But the problem is if an individual who has been exposed to AAV, and if they develop a very high titer of antibodies against AAV, then these patients have to be excluded from the treatment currently. And the community is still trying to work on this, try to get around the antibodies if the patient has it. So In addition to that, there is still room to improve the capsid and although AAV has been considered as a non-pathogenic virus, it doesn't mean that if injecting trillions and quadrillions of particles into a human body all at once would not pose any adverse effect. So we really need to keep that in mind, and we need to really design and evaluate each single vector very carefully and thoroughly before we put it into patients. So lastly, gene therapy has been traditionally considered to be a one and done treatment, which means you get an injection, you are hoping that you will get lifelong time therapeutic effects. However, the reality may not always be like that. So for example, if a quick example is if a DMD patient get a AAV treatment when he is a child, when he or she grows up, maybe a redosing is required because the body size all increase. However, at the current stage, actually redosing is not an option, at least for now. And the community, the whole AAV gene therapy community, we have been working on all of these challenges. So that is pretty much the challenges and the community is actively working on to address. But when we are considering using AAV to treat any specific disease, including OGT-XLID, there are some fundamental factors that we always need to think about. Firstly, there must be unmet therapeutic needs, where current treatments are inadequate or non-existent. So this is obviously a fact for OGT-XLID, and that is why we're having this meeting here today. Secondly, we need to have an AAV capsid that could transduce the target the cells efficiently enough. So in AAV community, we have many strategies to engineer the capsid that could cross the BBB maybe better, or maybe to achieve better tropism to specific cell types. However, before we do that, it is really important to firstly understand what the specific cell subtypes that the disease is mostly affecting. Once we identify the regions, the specific regions, the specific cell types, we need an administration route that would enable the delivery of AAV particles into the targeted place. Of course, most importantly, we need to identify the therapeutic gene and put it into the capsid to correct the disease, right? So in this case, we need to it's really important to understand whether the gene mutation is just a kind of loss of function or actually it is a gain of function mutation, because this will directly affect the gene therapy strategies when we design the therapeutic cassette. And what I'm showing you here on the screen is a therapeutic cassette for OGT proposed by Dr. Lester Rees on our team. So in this design, the OGT coding sequence serves as the therapeutic gene, and we have, because of the size of the gene, we actually have enough space to choose different regulatory elements to drive its expression. So as the previous presenter mentioned earlier, if we know OGT mutations affect some specific targeting proteins or some specific pathways, actually maybe we could also deliver or develop some other key genes, transgene, therapeutic gene to provide benefits for patients, especially if there is any concern of overexpression of the OGT may cause any toxicity in some specific cell subtypes. So lastly, once we have everything as far as the capsid, transgene, and a validated route of administration, then we need to assess the therapeutic efficacy in the cell model or animal model. I think this has been emphasized by the earlier presenters. So that is really helpful, and I'm just going to skip it. So with that, I would like to conclude my talk and send special thanks to Jude, Anna, and Lester, my current colleagues, and thank you for your attention. I'm very happy to take any questions. Thank you.
Yeah, questions, please.
Thanks for the nice overview, John Hanover, NIH. I'm curious about tropism. I mean, you know, that's big, always the debate, and we don't entirely know what set of neurons to target initially. What are you guys thinking about in terms of the tropism and which virus to start with?
Yeah, so that is a great question. In general, if we are not at this stage, if we're not quite sure what exact subtypes of neurons it may affect, then our target is just to get the virus cross the BBB and get into the brain. So we will choose a neuron, choose a capsid that is efficient in transducing neuron cells in general. And that, for that, like AV9 has been used for SMA, and there are many companies and the different teams that they are developing better capsid to targeting neurons. And I see you have a question.
I have an additional comment on the tropism. So tropism could be controlled by multiple mechanisms, right? So capsid is one of the regulators of the tropism. The second regulator of the tropism is a promoter. So, and it's really a combination of capsid and a promoter together that define the particular tropism. So you have seen on schematic from Stone that promoter had not been identified for this tentative expression cassette. And there are many reasons for that. In part, we have a lot of questions to the current community actually represented in this meeting, because in order for us to design the vector and test the vector, we need to know a lot of information. We need to know what are the cells and the cell types that are primary targets for this. Do we only need to target neurons, or we need to target all of the brain cells? We also need to understand the protein behavior. Is there an opportunity for cross-correction? Is the protein strictly intracellular, or there is some sort of exchange to the vesicles from cell to cell that could potentially help us to achieve a greater tissue penetrance, so to speak, right? Because imagine a situation where we could transduce 20% of the cells, but if there is a cross-correction mechanism that goes in there, then more than 20% of the cells could potentially be affected by the expressed proteins. So we also need to understand, is there value in this, for example, decreasing expression of OGA, because we could incorporate something that is anti-OGA microRNA into the same construct and decrease the expression of the counterpart. So this is a tentative idea. It's not a fully formed idea. And what we're looking from this community, if to have an assessment whether gene therapy is worth pursuing in this particular disease. And if it is worth pursuing, then we need to better understand some of those details that I mentioned earlier.
Thank you, Anna.
Gülçin Pekurnas from UCSD. I also want to raise another important point about the delivery of OGT, subcellular targeting and protein turnover. Because based on the design, this is cDNA-based delivery, right? We know the alternative splicing. We also know OGT is highly enriched at presynaptic, postsynaptic sites, and the protein turnover in mouse brain in a couple weeks. So how are we going to ensure that the cDNA is going to be properly expressed and delivered to these subcellular regions in long neurons? Maybe we also have to think about the translation and local regulation, which requires subcellular studies.
Thank you. That is a very fair point. And yes, we need to learn more about the splicing, whether the protein, like Anna also mentioned, how the protein behaves. Like they need to, whether the protein actually, after splicing, do they actually work as a monomer or dimer or things like that? It's all very important to learn. And when we design the cassette.
So for the alternative splicing isoforms, depending on the composition of the alternatively spliced proteins, we could create artificial short introns that actually allow for expression of multiple splice isoforms. It's a little bit more sophisticated approach than the traditional cDNA-based gene therapy. So, but those things have been done before. So, and again, we need to have more information in order to better understand, if there is a particular isoform that is a key one, or there need to be 2 or 3 key isoforms that need to be co-expressed at the same time. So in regard to the biological function of the protein, that will have to be confirmed experimentally. You're completely correct. Sometimes delivering cDNA into a target cell is not enough because we have an artificial promoter, it's a synthetic construct, and we are not reproducing the biological pattern of expression. So, and many times what we have to do is we have to go back and actually evaluate the protein composition. Sometimes we need to tweak. If the protein has a pre-pro protein composition, we need to tweak the pre-pro protein cleavage signals to ensure that we generate in the correct final product that is expressed from the vector. So it's not just the vector engineering, sometimes it's also protein engineering. So that's why approaching this as a community with the experts in the disease and expert in the biology of this protein behavior and linking it into the therapeutic cassette design is critically important because it's not enough to express the protein. What we need to do is we need to express functional protein in the right place. But there are certainly molecular biology tools that we could employ to achieve that.
Thank you, Anna.
So I need to, so Judy Fridovich, Kyle Emory University. So I've seen 2 kinds of pedigrees shown to us today. One of them shows inherited X-linked mutations where ostensibly a carrier mom is healthy and passes it to a son who has the condition. And then I've also heard that, that many families have a de novo mutation, in which case I'm not really sure that we know that this is recessive. If it's a de novo mutation, it shows up in the affected child and is not found in a carrier mother, then it's possible that some of these mutations, in fact, may have a dominant, mode of action, in which case giving back the wild type, the wild type gene product may not be a clean fix. So if for those that are inherited and a carrier mom is healthy, that's a very good sign that it's recessive. But in conditions where it's de novo, I think you need to show that it's recessive before you can assume that giving back the wild type, because it may not be a loss of function, it may be a change of function. And that's not something that's always overpowered by giving back the wild type gene product.
I can chime in here too. Sorry, Son. So we have cases where we develop vectors for diseases which are definitively dominant negative diseases, and the way how we utilize vector design there is we use the knockout and replace strategy. So I actually posted that question in a chat earlier about whether the mutations are dominant negative sometimes, and if it's known about this particular disease. Because we could create a vector with a dual composition, again, it's a single vector, where we have a microRNA that targets endogenous allele, and we are doing a knockout and replacement strategy. In fact, we're about to submit a single patient IND for an ultra-rare disease, which is a dominant disease where we utilize that particular strategy. But you're completely correct. Understanding the nature of the mutation, whether there is a gain of function, and what is the exact function that had been gained by that mutation is critical in the vector design.
Andrew? I was just going to say, we've also heard of affected females, and we've heard about skewed X inactivation, and knowing which X is skewed, and all of that can also be helpful in determining this for affected females.
Yes, absolutely. Because we have the case of Victoria, who exhibits some of the symptoms, right? So, and she's a female. So, and she only has one allele that is mutated. So something is going on there clearly. And I think it would be critical in the case of Victoria to identify if that mutation has a gain of function or it could be a dominant negative mutation.
We need to move on. Liujiang, Anna, thank you so much. I will just add to that Anna's team is working on designing a capsid for OGT, and actually, like, we would need to connect Anna and her team with the researchers who are working on that, just to get more information, and we're absolutely happy to make those connections and introductions, because Anna and Liujiang are asking questions about, like, hey, what about like this, or what about that, and we just need more better answers for that. Thank you, and we'll move on to Sandra. Sandra is director of the Office of Orphan Product Development in FDA. Sandra actually joined from the beginning of this conference. Appreciate your time, Sandra. I know that when we last met, you were very kind to share more details, and just show commitment to families like ours, and the parent organizations like ours. So please welcome Sandra Retzky from FDA.
Hey, everybody. Can everybody hear and see okay? Hey, can you guys hear and see me okay? Yep.
Yes.
Okay, super. So I'm Sandy Retzky. I'm the director of the Office of Orphan Products Development, and I'm so pleased to be here with you today. Thanks so much for reaching out to FDA and asking us to join. We're really delighted that you did. First thing I'm going to do is talk about a couple of definitions so that we're all, at least from a regulatory perspective, we're all talking about the same thing. Oops. There we go. What is a rare disease? So I'm going to define rare disease, and then I'm going to define orphan drug. So for the purposes of regulatory decision-making, a rare disease is defined by statute. It's been defined by Congress. And it's different for drugs and biologics versus devices. And what you see here on the slide is the definition for drugs and biologics. It's a prevalence of less than 200,000 people in the United States. And for devices, it's an incidence of less than or equal to 8,000 people in the United States. So that's rare disease. Orphan product is a product that demonstrates promise for the diagnosis, treatment, or prevention of a rare disease or condition, and it can include drugs, biologics, medical devices, and even medical foods. And from our standpoint, from a regulatory standpoint, rare diseases present many challenges, and it's been great listening to everything you guys have been talking about today. You've talked about a lot of the challenges, but from a big picture perspective, one of the big challenges is a genotypic phenotypic heterogeneity. And I kind of heard some of that today. So you might have different phenotypes, genotypes, but people express the same phenotype. So the underlying genetics may be different among a disease, but they all express themselves physically, manifest themselves physically the same way. That makes it difficult for drug development when that happens. It's also, because they're rare diseases, it's in a small population. And so it's tough to do product development in small populations, particularly when they're widely dispersed. So often we need to have a lot of coordination, not only in the US, but internationally, worldwide, when we're trying to do studies for product development in rare diseases. And then the natural history of the disease may not be well understood. And I'm hearing some of that as well. And one of the things that we want to do when we're developing products for rare diseases or any disease, including common diseases, you want to know that it works. And in order to know whether a drug or a medical product works, you have to compare it to something. So that's why understanding natural history should be really high on the priority list for drug development for rare diseases, because sometimes we can do a lot once we know the natural history, the natural course of that rare disease. Sometimes we can even use that natural history as an external comparator so that we don't have to have a placebo trial necessarily. And then and I heard somebody talk about endpoints today, and I did put a resource about biomarkers and endpoints into the chat. Endpoints can be really difficult, particularly when a disease is not well understood. So an endpoint, and I heard somebody talk about this, from FDA's perspective, an endpoint, a primary endpoint, is the way a patient feels, functions, or survives. That's not the same as a biomarker. A biomarker is an intermediate endpoint. And I heard somebody rightly say there has to be some nexus between the biomarker and the disease such that one would believe that if there's a change in the biomarker, it would naturally follow that there would be a change in the disease as well. But for traditional drug approval, what FDA looks at as a primary endpoint is how someone feels, functions, or survives. Okay. The next thing I'm going to talk about are incentives. I want you to know that I'm not in a review division. I have been in a review division where we've reviewed products. I've reviewed lots of gene therapies, cell therapies, tissue therapies. I'm actually in an office that's in the office of the commissioner. And one of the things my office does is provide incentives for product development in rare diseases. There are 3 medical product centers at FDA, one for devices, one for drugs, one for biologics, that review product applications for licensing products to go into commercial use. That's not what my office does, but there's a lot of interplay between my office and the review divisions. We go back and forth all the time, and we know each other very well. But what I'm going to talk about is incentives that are in the Office of Orphan Products Development, which is in the Office of the Commissioner. So, incentives. These incentives, the first ones I'm going to talk about, and by the way, this is a very abbreviated presentation today because I know we only have a few minutes together. I'm going to talk about orphan drug designation, orphan drug exclusivity. That's one type of incentive. I'll also just touch on rare pediatric drug designation and priority review vouchers. That's another type of incentive that I thought you'd be interested in. I'm not going to talk about devices today. And by the way, when you think about devices, in vitro diagnostics are a type of device, and that is also something FDA approves. But I'm not going to talk about them today because we have such a short time together. So the first program you might be familiar with, it's our biggest program, it's our longest program, is the Orphan Drug Designation and Exclusivity Program, which was started in 1983 from the Orphan Drug Act. It's a very important piece of legislation. Before the Orphan Drug Act, there were very, very few products for rare diseases in the United States. And there are 3 main incentives for orphan drug designation. These all come prior to licensing or marketing application for commercial use of the drug. One is pre-development. It's tax credits to defray the costs of qualified clinical trial costs. So that's one. A second is waiver of a marketing application fee, which I put for you this year for fiscal year 25 is quite hefty. It's $4.3 million. So what that means is when an investigator or an investigator sponsor is ready to have FDA look at their file for commercial approval, they have to submit their file to FDA for our review. And it also comes with a licensing fee, a non-refundable licensing fee. And that's $4.3 million in fiscal year 25. But if the product has received orphan drug designation sometime prior to the market application, that fee is waived. So it's a nice incentive. And the big one is 7 years of marketing exclusivity. Let me explain what that is. So marketing exclusivity, think of it like a government-sanctioned monopoly. It's very unusual. FDA will not approve another same drug, meaning the active moiety or molecular structure, for the same use or same indication by a different sponsor for 7 years. That's a great incentive. Let me give you an example. So if drug A has orphan exclusivity for treatment of cystic fibrosis, FDA could not approve another same drug A for that disease for 7 years. However, FDA could approve another drug that's very similar for cystic fibrosis, as long as it's not drug A. And it could approve drug A for other rare diseases as well. So it's a kind of a narrow monopoly, but it really is quite incentivizing to commercial sponsors. Very briefly, making a designation request. So that has to be, as I said, submitted prior to marketing application. Need to do basically three things. One, describe the disease or condition. Two, provide a population estimate, and you have to support that with some evidence that it is what the sponsor says it is or what you say it is. And that evidence often comes from literature. It can come from other sources as well. And then it needs to be supported with a sufficient scientific rationale as well. We need to understand why the drug that the sponsor is submitting to us for orphan drug designation has the medical plausibility to treat that disease. So three things. And just briefly, sufficient scientific rationale, evidence that the drug holds promise for being effective, and that data often comes from clinical data, and it can be case study reports, it can be literature reviews. It can come, if there isn't any clinical data, it can come from animal model data, and I heard someone talk about animal models earlier today. And on rare occasion, we will also accept in vitro data only if no animal data or human data exists. The important thing is to know that you can get orphan drug designation without having an adequate and well-controlled trial. So it's a much lower threshold to get before submission of a marketing application. The threshold, the scientific threshold for getting approval, FDA approval of a product to put into the marketplace is much higher than this. But this is a good start. It gives some nice incentives to the sponsor, financial incentives. I also think it gives some important validation for follow-on investment, and whether that comes from friends and family money, angel investor money, venture capital, pharma, biotech. Having orphan drug designation can be really quite valuable. I said I'd very briefly mention the Rare Pediatric Disease Priority Review Voucher Program. The purpose is to incentivize development of therapies for rare pediatric diseases. And unlike orphan drug designation, where you can get some pre-approval incentives, there are no pre-approval incentives here. There is no tax credits. There is no waiver of a marketing fee. But upon approval of something that's been designated a rare pediatric disease application, the sponsor is eligible for a voucher that can be redeemed or sold so that it can be applied to a subsequent different application. And that application, whether it's for a common disease or a rare disease, will get a priority review. And that means the review clock goes down from 10 months to 6 months. And for many sponsors, that 4 months, it might not sound like much, it's very meaningful. And for some companies, they sell these vouchers. It's sold on a private marketplace, can be sold to a person or to a company. And they typically, the last few vouchers that have been sold go for about $100 million. So I think of this as a back-ended milestone payment that is not being borne by the government. It's if somebody's going to sell a voucher, it is a private party. It is not the federal government that is providing this milestone payment. The only time we get involved is on the subsequent application. Our employees have to provide a much faster review clock. That's the only part of the voucher program where the federal government has any kind of a tax on it, so to speak. A designation, a rare pediatric disease designation, can be obtained anytime prior to approval of a product, but it doesn't necessarily guarantee approval. So you can submit one anytime during development, but it is not a piece of paper that says you're going to get approval. This is the second part of my presentation. Very quickly, I want to mention some resources that are available when you're considering doing product development for rare diseases. And there's quite a few. First, my office has a grants program, and so I want you to know about that. We have a clinical trial grants program. We have a natural history study grants program. We have a FDA Rare Neurodegenerative Disease Grants Program, which is relatively new. So the Clinical Trial Grants Program and the Natural History Study Grants Program began with the Orphan Drug Act in 1983. The Act for ALS, which was enacted in 2021, started this new grants program called the Rare Neurodegenerative Disease Grants Program. We just had 2 receipt dates in October for clinical trials and another one for the Rare Neurodegenerative Disease Grant Program. We'll have a natural history study receipt date probably in February of next year. And so we've got a whole bunch of applications that came in for clinical trials and rare neurodegenerative diseases. We'll be reviewing those in the early part of next year and then making awards before the end of the fiscal year next year. One thing that I think is really nice about this grants program, it has a long history of being quite successful. I was really surprised when I came to this office how many products have been approved with this small grants program. This is not like the NIH, some of the NIH programs, which are quite large. You know, the Orphan Products Grants Program has about $25 million in it. That's it. And so when we give a grant, it tends to be much smaller than what, say, NIH might give. It also tends to be much earlier in drug development. And like I said, it's had a really good success rate. So and sometimes it's the only grant that early stage companies or an early stage sponsor gets. And we've seen a number of times where because they get a grant from FDA, other money follows on. And I think that's great. That was the purpose of establishing this grants program. Also wanted to tell you that we have expedited programs to review products and to give sponsors help. So if there's anything you're interested at FDA, one of the things I did to put that, I put something about biomarkers and guidance, our guidance about biomarkers in the chat. All I did was go biomarkers guidance FDA.gov, and I did a Google search and found it. So if you're interested in expedited programs or even disease-specific programs, just write whatever you're interested in, say epidermolysis bullosa, which is a rare disease, guidance, FDA.gov. And if there's a guidance, it'll pop out that way. But these are our guidance programs our expedited programs. We have fast track, breakthrough designation, accelerated approval, priority review, which I've already mentioned, and rare regenerative medicine advanced therapies, or RMAT. So one thing I will mention is accelerated approval. These are all spelled out in this document if you want to see them. But what's interesting is the accelerated approval. That is not a traditional approval pathway, and usually approval is done on a biomarker without an endpoint that shows how a person feels, functions, or survives. We understand that sometimes in order to know whether you have improvement on a primary endpoint, like a patient-reported outcome or a clinical reported outcome, it could take 5 years. That's too long. So we will allow accelerated approval and approval on a biomarker, but then the sponsor has to do a confirmatory study to convert that accelerated approval product into a traditional approval. So anyway, I wanted to point this out to you because this is a really good guidance to start with, as is this. So guidance documents represent FDA's current thinking on a topic. They don't bind FDA or the public. And certainly you can use alternative pathways or approaches as long as it satisfies the requirements for the statute and regulations on approval. And I had mentioned the Google search. But the other thing I wanted to mention that's really good are meetings with FDA. We have a lot of meetings that we offer to give guidance very early in development. And this is a nice guidance document to look at the different kinds of meetings that FDA can offer sponsors or groups like yourself. In this particular one, there's another one on medical devices as well. So we have PDUFA products, are prescription drug user fee products. Those are things like small molecules, large molecules. I heard about gene therapy. Those are PDUFA products. But there's also medical device products that we offer advice on as well, including in vitro diagnostics. So last thing I'll say, I'll just mention that please save this date. We have FDA and NIH will be having a combined Rare Disease Day event during Rare Disease Week, which is the last week in February. This year it's February 27th and 28th. So please save the date, and we'd love for you to come. We will have both virtual attendance as well as in-person attendance at the Natural Auditorium. So as a closing thought, incentives and resources are available from FDA. We're very interested in rare diseases. More than half of the drugs we approve now are for rare diseases. So please contact us early, and use these resources to help you. Thanks. Be glad to take any questions.
Oh, we've got a couple of them. So let's start with Daan.
That was a really informative talk. Thank you very much. To what extent are those funding sources available for non-US based groups or sponsors?
Doesn't matter. Doesn't matter. It can be foreign, but I'm glad you asked. It's for public entities, private entities, US based entities, foreign entities. Doesn't matter.
Okay, thanks.
Sure.
I also see there's a question from Joe. How might the Rare Disease Innovation Hub, the new hub that was recently created at FDA, be folded into the ongoing rare disease efforts at FDA, or too early to know yet?
Well, so thanks for that question. The Rare Disease Innovation Hub is very new. And I think it was just last week, maybe the week before that, the director of that program was named. But the purpose of the hub is really to bridge between the Center for Drug Evaluation and Research and the Center for Biologic Evaluation and Research to make sure that the programs that go through those review divisions are aligned in terms of their endpoints and making sure that the patient voice is being heard when these products are being reviewed. It's a way of aligning FDA and what the review divisions are doing, and also giving more transparency to the public. So that innovation hub is really meant to have a way to see into what we're doing, to make sure that what happens in one center is the same experience that you have in another center, to the extent that can be done. You know, sometimes things can't completely align for the same disease, because what might be needed for gene therapy is different than you need for a small molecule. But at least it's a chance for the public to hear the discussion and also ask questions and have their voices be heard. So it's not it is not an approval or decision-making body. So I think that's an important distinction. It's much more about having the public see what we're doing and maybe trying some new things in terms of innovation in clinical trial design, for example. So I hope that helps. But it is early days.
Thank you.
Yeah, thank you. I also have had a question about this Innovation Hub meeting. So there was recording for 3 hours when different people, parents, companies talk about issues that they see in FDA, and they come up with recommendations and ideas how to make it better. And that meeting was back in October, and I was just curious,
Yes.
are there any outcomes from that meeting that you might share, or it's too early? I mean, because one month sounds like a lot to us. Maybe in your world, you measure it in decade.
Well, we are the federal government.
Yeah. So my question, any actual outcomes from that meeting? Because there were a lot of really good ideas.
Yeah, no, there were. And the meeting that you're talking about occurred on October 16th. And so if anybody's interested and you would like the transcript of that meeting or recording of the meeting, just go Rare Disease Innovation Hub, fda.gov, and that page will pop up. But right now it's being stood up, and a charter is being developed. And so right now there's no outcome yet. And in rare diseases, I don't think we can go fast enough. And so I understand that a month has gone by, but I think in all likelihood, it'll probably be the early part of next year where the first meeting takes place.
Got it.
Makes sense.
And a quick question,
Sure.
one more question. For pediatric voucher, correct me if I'm wrong, but it was extended till like November or something. What are the future plans for this program?
It was extended to December 20th.
December 20th.
And so, yeah, and thanks so much for bringing that up. I had to go kind of fast through my presentation, but what the question is about is the rare disease pediatric review voucher program was supposed to sunset on September 30th of this year. And so Congress, when they did the continuing resolution and passed the budget to go to December 20th of 2024, there was a rider on it that extended the priority review voucher programs for rare pediatric diseases to the same time, December 20th. And so, we hope that Congress will reauthorize the program fully, for at least another 4 years or 6 years. Congress, when these programs first start, they often will have a 4-year, 6-year time frame, and they're renewed a couple of times to determine whether or not the policy is effective or not. But given what's happening in Congress, I'm not sure what will happen on December 20th. They could fully reauthorize it. They could give another short-term extension of 3 months, or the program could lapse. So there's some uncertainty there. And it's not up to FDA. This is Congress. It's Congress that had decided. Remember, I said what a rare disease is: 200,000 people or less. That's from Congress. Congress gives us the scientific threshold to approve drugs. We use our judgment to decide whether or not it meets Congress's threshold. And the same with these kinds of programs. It's up to Congress to decide whether to reauthorize them, or to not reauthorize them, or to make them permanent. So I hope that helps. But it's not FDA. We'd like to see it continue.
Right, everyone? We're starting our last session.
All right, everybody, please take your seats. We're going to get started.
Oh, you got it up there.
Okay, y'all. So I'm just going to give a brief introduction to the workshop while all of y'all stand up and rub yourselves, because it's kind of cold in here. Don't rub other people. All right. So that's me. Sorry. So part of this is kind of a brainstorming session, and then we can start thinking about things like a white paper and funding efforts of how we put teams together and we get things done. Thanks to the many of you who filled out the questionnaire. So I kind of went through the questionnaire, kind of tried to unpack it a little bit. And what you really see, and I think what we've already seen for a few hours today, is really common ideas, challenges, logical steps forward clearly emerge. The one thing and I'm not trying to pour cold water on anything, but I think the basic scientists were all kind of in agreement that there's just not enough preclinical data. Like, there's no way we can quite move forward yet. We don't know that expressing wild type OGT in any system will fix anything. We don't have that evidence, right? And so, so what do we need to do, right? So what do we need to do? Because we want to get there, right? We want to get to a therapeutic. That's the whole goal of this meeting, right? Is what's the best way forward? And so, right, I'm kind of beating a drum that's been beaten all day, and that is we need a reliable biomarker of disease. And we need it for a lot of different reasons. We need it for a definitive diagnosis of novel mutations and variants. And it sounds like Dane might actually have something. I'm totally looking forward to seeing that poster at SFG. We need it if we're going to do gene and drug screening, right? I mean, my lab does, CRISPR screens, right, on other diseases. I can't do it here because I don't have a biomarker to read out, right? And so I need something to read out, right? For following the efficacy of any therapeutic approach, we're going to need a biomarker, right? And so this is going to come up. And so, how do we currently approach these new mutations and variants, right? Dane's getting a call once a week in Europe. I'm getting a call probably more like once every 2 weeks in the US, right? And so we've got to figure out, wow, my voice doesn't carry? That's hard. Sorry. So we've got to figure out, how to diagnose these patients in a logical manner. The good thing we have going for us, right? I don't know if everybody's aware of this, but right, in humans, there's roughly 220 glycosyl transferases. OGT is by far, it's not even close, the most conserved vertebrate glycosyl transferase by sequence, right? And that's been demonstrated by multiple groups. So and also in the human population, we just don't see a lot of SNPs, right? They just don't exist, right? And so when you take, the 10,000 healthy human exome database, there's nothing there. It's just wild type. It's just one sequence of OGT, right? For the most part. And so when we see a mutation like here, right, this red alanine, you see every sequence. We're going all the way down to C. elegans and Drosophila, and we're hardly seeing any pickup of any mutation, right? Highly conserved gene. So that's one of the first things we do, right? Does it occur in an exome database, even though that exome database doesn't have that many variants? Is it conserved across organisms? Is it predicted to be disease-causing in silico? And then we do some 3D modeling, right? And 3D modeling's gotten really powerful now, right? Won the Nobel Prize this year for being able to do things in silico, right? It's really powerful. And, right, we can look at that. But that's kind of where we are, right? And we go, okay, it seems to pass all those criteria. I guess it's probably causal, right? And hopefully, if we have some family history, hopefully, it's been passed down and all the uncles that are unaffected don't have that allele and the affected uncle does have that allele, right? You start building family trees and you believe it. But again, if you have a de novo mutation, yikes, right? Like, how do you know? So what else do we need reliable biomarkers for, right? And I mean, how do we get there? So maybe it's RNA-seq, maybe it's OGT chip-seq, maybe it's O-GlcNAc proteomics. Man, is there a single O-GlcNAc protein that's just not getting O-GlcNAc modified properly, right? It's possible. I can tell you in L254F, we went 850 sites deep, and the correlation was 1. Right? It's a perfect line, right? There wasn't anything more than 10% off the line by O-GlcNAc proteomics. And 800 sites is pretty deep in our field, right? I mean, I realize there's 8,000 sites, but at least for the top 10% of sites, there's no differences, right? And that's, of course, for a TPR variant. The OGT interactome, right? Whether you're doing that by IP or bio-ID, whatever your favorite approach is, maybe that's where we're going to find it. Right? Maybe all the TPR variants don't interact with, some protein. Or maybe it's going to be a metabolite or something other, right? Because one of the things we're all focusing in on is the fact that something's going on, and it's a neurological disease. That doesn't mean the defect's in the neurons. It could be in the glial cells. It could be in the liver, right? I mean, everybody, right, at least the scientists in the room, right, appreciate PKU is a neurological disease, right? That's where the phenotype is, right? But you fix it, all you got to do is replace that enzyme in the liver and you fix the disease, right? It's right because things don't get expressed, secreted properly. We know O-GlcNAc affects all sorts of adipose tissue. Adipose tissue is an endocrine tissue. Maybe something's going on there, right? We don't know, right, exactly what we have. And so the question becomes, what systems do we look at as well, right? We have a lot of them. We talked about a lot of them today, right? I'm not going to go through all this, but right, fish, flies, C. elegans, right, in vitro and cellulo in vivo. And I think from the worksheet and from my own lab, and I think Dan's lab and everybody who works in this field, the answer is yes, 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 right? We need to be in all of these systems. We need to be doing this with as many variants as we can handle, right? Because we've got to find common phenotypes if we're going to move the field forward. Or we just have to get more information, right? If we're going to build this, semi-clinical trial with glucosamine, it would be great to get 10 variants, that all those variants have a KM mutation in sugar nucleotide binding, right? That's going to be much more successful, or has the opportunity to be much more successful than just 10 random patients, right? We might need domain-specific biomarkers, right? And maybe OGT would benefit from SMRF, right? Which is this idea of just mutagenize the whole thing and figure out everything that causes a problem, right? Anything that lowers kinetic parameters. But again, even with SMRF, it's good that you need a biomarker, right? If you're going to do SMRF, you need a readout, right? So we use SMRF in my field. The other half of my lab works on congenital muscular dystrophy because we have this great readout of this antibody that only binds the cell surface when that glycan's built exactly right, right? And so we can mutate all of the enzymes in that pathway and go, oh, look, mutations in these domains cause this problem. We don't have that right now, right? So we need a biomarker. We need it for performing genetic and drug screens, right? CRISPR knockout, CRISPR-I, CRISPR-A screens, right? We would all love to run these screens, right? FDA approved drug screen. But the question is, what's my marker? What am I, right? My laboratory, I'm sure Dan's laboratory, I'm sure multiple people in this room's laboratory, right? CRISPR knockout I and A screens are routine these days, right? But you got to have a good readout, right? You can't do a screen unless you have a good readout. So we need that biomarker. Of course, we also have to ask ourselves what variants we're going to do this on, right? CRISPR knockout screen is going to take a fair amount of time and maybe $15,000, $20,000, right? Once you get all the sequencing and everything done and you actually do all the validation, making individual knockouts, right? That's a really expensive screen. I don't think we want to do that on 80 variants, right? It's just not, it's not useful, right? It would cost over $1 million. So what variants, what systems? These are the questions we got to be asking ourselves. And we got to be able to follow efficacy of any therapeutic approach. We've already started talking about these, right? Substrate enhancement therapy, OGA inhibition, right? Eli Lilly's got a nice drug. It's gone through phase 1 clinical trials. ACE neurons is through phase 1 clinical trials. Merck Electos, it's through phase, all three of their drugs are through phase 1. So they're past safety, right? But can we repurpose those, right? But again, if you're going to repurpose them, for what purpose, right? What are you going to measure, right? Because in a TPR mutant, there's nothing wrong for the vast majority of TPR variants, unless they're destabilizing, which some of them are. There's no change in O-GlcNAc levels. So how does elevating O-GlcNAc levels by inhibiting the OGA, how's that going to help us, right? It's not clear how that would help. Right? And again, I'm not trying to rain on parades. I'm just trying to talk about the limitations we have that we've got to figure out. We still need more preclinical experiments going on, right? Gene therapy, right? Gene therapy would be amazing if it works, right? The question is, are we there yet? And I don't even think we're there yet, right? I don't think we can propose doing gene therapy until we demonstrate that overexpressing a wild type OGT is going to help, right? I mean, I can go in with CRISPR-Cas9, I can fix the mutation in the cell line, and of course that helps my cell line, right? My cell line behaves just fine now, right? But, right, we can't do that in humans. Here we're talking about AAV therapy, we're talking about just overexpressing wild type, right? So the mutant's still going to be there. If there's irreversible damage, it's still there, right? We just don't know enough to do that yet. And where would we try to target that gene therapy? It's not clear at all. So, open discussion. Get back to earlier slides. And really, hopefully the next 30, 45 minutes, we can focus on how do we, what are our priorities moving forward? What does the field have to have, right? And this is why we have people in this room who are not necessarily experts in O-GlcNAc, but are experts in moving fields forward, right? And have done this for other diseases, people like HUD, people like Judy, right? Who don't particularly work on OGT per se, right? But they know how to see a field advance, right? And can we start thinking about funding opportunities for team science? It sounds like maybe we heard about one just a few minutes ago, right? Isn't bound by the U.S. border, which is kind of nice, right? And then there are U.S. border, unfortunately, limited ones, right? Like, program project grants, right? Program project grants can really move fields forward. And so, are we interested in doing that as a group? So I stole this slide from Stephanie and Natasha, right? Just to get back to thinking about this idea of how we get towards biomarkers, just to open up the discussion. And that's what I'd like to do now, is actually have an active discussion.
Thanks. That's a wonderful overview, Lance. And what I'd like to suggest is that I think there are clearly many areas in which this condition, there are too many unknowns here, and I think that things need to happen in parallel, not in sequence. So if we imagine, okay, somebody's going to find a biomarker. Okay, it's going to be a beautiful biomarker, and somebody's going to show that gene therapy works in a mouse model or whatever. Eventually, when the field gets to a point where you want to try to do gene therapy, or I'm sorry, you want to do a clinical trial, you're going to need the natural history information because you're going to need to design that clinical trial. So I personally would recommend that while one team of people or whatever are working on the biomarker or the basic science, the preclinical models, that the natural history study should be starting like ASAP. And I guess I would volunteer to help with that if that would be useful. I've done that for more than 30 years with classic lactemia, and we've learned a lot, and it's been very helpful. So I think that that is something that I think your community needs, and it needs to be happening in parallel with all of the basic science that obviously also needs to happen. The other thing I just wanted to say is that I know that some people would say that it's okay to do a clinical trial without a placebo group if you know enough about the natural history of the untreated condition. And I just want to throw out there, I've interacted with a lot of families whose kids have participated in clinical trials. And as any parent knows, child development is not uniform and it's not linear. And so I have parents who will say to me, oh, my kid is doing so much better, and I'm sure it's because they're taking this drug. And then I have other parents who tell me, oh, my kid is doing better, and I say, oh, do you have any idea what's going on? And it's like, they got this new teacher, and the teacher just is really motivating them. And it's like sometimes kids who were struggling start doing better. And I have a clinical colleague who does gene therapy, he does clinical trials for all sorts of diseases. And he said one of the first trials he did, every single parent swore that their kid was doing better than they had ever done. Half of them were on placebo. Right. Okay, and he said it was just must be his winning personality. So I think placebo hope is a really powerful thing. And so when you're dealing with squishy things like child development, I think you need a placebo group.
Thanks, Judy.
So, Lance, a couple of things come to mind.
It's John Hanover.
Oh, sorry. Yeah, I got to do it down here. First, knowing the NGLY1 community pretty well, and what drove biomarker success there is a biochemical mechanism. We know something about the biochemical mechanism here, but I'm curious, maybe we have many members of the community here, whether genetic modifier screens are in progress. I mean, genetic modifier screens take many forms, but they might be the quickest way to get systemically to targets. Because, for example, Clement Chow showed that, in GLY1, his fly mutants were completely dead, but then he could recover 100% viability with a genetic modifier. And I don't know that, I've thought a lot about it. I don't have the wherewithal to do it, but I think in this room we do. And that would, I think, give us a clue as to what street light to look under for the keys that we lost, right? So and the other thing is, we should reach out, in my opinion, because I'm ignorant of what's known about X-linked intellectual disability generally, for biomarkers that may already be in use for some of the more common X-linked intellectual disorder.
Michael, on Zoom, please feel free to unmute yourself.
Hi. Yeah, so I wanted to touch on just agreement with, I've been a part of a number of programs that have gone from where you guys are at to, the human clinical trial phase, particularly in gene therapy. And so I completely agree. You need to know more about your animal model. You're going to need your animal model kind of tier. You're going to need your natural history tier, which I completely agree should start like tomorrow, because you will, first of all, you don't know what your clinical trial terrain is going to look like in terms of the types of designs people will decide are standard by the time you get to the trial phase, but you will never regret knowing more about your patient population, or as much as possible, especially for a 5-year period, that your trial might need to go for, right? So you can see where things kind of are headed. And I would also say that, this idea of moving in parallel is really important, because even if you're just getting the mouse model up and running and you're just sorting through what the AAV constructs are and things like that, if you can use those as tools to interrogate each other, if you can look at the impact of what overexpression of your wild type protein is by using gene therapy as kind of the tool to manipulate that might save you a little bit of time if it's feasible. And obviously, you need to have this kind of siloed in a certain way. But it's, definitely separating your people out into teams. I wouldn't worry too much about the specific technique yet to look at your biomarker, but rather the protein or the RNA or whatever itself, the thing that you use to find it may not be what you use to make it a validated assay at the end of the day. So you don't need to make all those decisions right now.
Thanks. That's really helpful. Yeah, I mean, the natural history, at least the way my laboratory is handling that right now, is everything is getting funneled to Eva and actually Andy, right? Part of the Frontier CDG program, which is being incredibly renewed, hopefully. Right, it's in renewal, hopefully. It's submitted. So what we're trying to do is get everybody going to the, right, people can go to whatever physician they want. We want that physician communicating with Eva and Andy so that we can get natural histories, right? That makes sense, right?
Yeah, and I mean, I was at meetings 10 years ago that were just like this and were no larger than this one, with massive differences in the ability to treat the diseases and some of the muscle diseases that we were working on back then. And so, this is exactly what you guys are supposed to be doing right now. But don't be paralyzed by the number of choices that there are to be made. If, you're focusing on the right things in terms of sorting out targets and learning more about the disease and models itself, because those are the kinds of things that are going to be the foundation of anything you're bringing forward.
Thanks.
Hi, this is Mike Boyce from Duke University. I wanted to make this is sort of building on a couple comments that have been made about parallel approaches. I wanted to put in a plug for model organism genetic screening and maybe drug screening in model organisms. I feel like, the biomarker work and natural history is obviously key. A virtue of model organism efforts is that you don't need to know that information yet. If you have behavioral or survival phenotypes in C. elegans or in Drosophila models, then that allows suppressor enhancer screens with genetics, with drug exposure that are agnostic to underlying mechanism. Also, it has the potential for revealing things that manipulate compensatory pathways that aren't part of the disease mechanism but are useful therapeutically. That doesn't rely on a molecular biomarker necessarily. If you have a behavioral assay or a survival assay, it doesn't rely on the patient natural history work to have been completed. So those would be hypothesis generating experiments, I think. Something that works in a worm or a fly, of course, doesn't always translate to vertebrates. But I think
but other times it wins you the Nobel Prize, right?
Other times it wins you the Nobel Prize. Yeah,
right?
and we were hearing earlier from Daan, for example, that some of the some good hypotheses about underlying cellular mechanisms could include highly conserved processes like synaptogenesis or synapse firing, basically. So that can be studied in C. elegans.
That's a great idea. I mean, loss of the beauty of C. elegans, right? I mean, John showed this, and we played around with this
Yeah,
a little.
John has a model.
Right,
You
it's
could
not
be the
lethal.
next Bob Horvitz for the
Right, but it has a lifespan phenotype, so all you got to do is screen for your worms to stay alive, right?
Yeah,
At a certain
right.
number of
I mean,
days,
death
right?
is the best phenotype in a genetic screen like that.
Right.
So, you know, like,
exactly.
yeah.
Live dead is a really powerful screen when you're working with C elements.
So we have a paper coming out in eLife shortly that shows a sleep defect in flies that have OGT-CDG mutations. I agree that that's a great organism to do genetic or drug screens in, but I think the problem that we're facing is that different variants have different effects in different model organisms, and so then what are you screening for, and how do you know that the result that you find is applicable to all of the variants that we know in humans?
Yeah, no, this is a great point. I mean, we have this, even in cells, like, you know, one cell, right? Just a cell line. Variants have phenotypes all over the place, right? Just in their growth rate, in their O-GlcNAc levels, in their OGA levels, in their RNA transcripts, right? So trying to find something common is going to be a major challenge. It just gets more complicated as you go to a more complicated organism.
Yeah, I mean, with CRISPR and cells, we've recently been asked by referees to do whole genome sequencing in our cells, and I think this is absolutely essential. Is that CRISPR isn't the golden bullet that it's
exactly.
presented to be. So there are, you know, we have definitely got cells and flies that are genotypically supposedly completely identical, but phenotypically very, very, very different, and so that hints at all kinds of nasty background CRISPR scars that contribute. Yeah.
Yeah, I mean, this is the thing that concerned us the most about L254F. Human fibroblast from the patients had a defect in OGA. We make the RUES1 cell lines using Cas9, and there's no defect in OGA, but there's clearly, I mean, you know, we sequence, it's defective, it's got the L254. That's just switching cell lines, right? That's terrifying.
Can I make a quick follow-up comment? So as I pass the microphone to Andy, sorry, I completely agree with Don about the necessity of looking at many variants. It's a lot easier and faster to do that in model organisms than in mice, right? I think that's another important virtue.
Yeah.
There's comment from Hudson, who's been up for a while now. Please go ahead.
Yeah, hi guys. Hey, I'd like to second what Judy said, that it's really important to have controlled trials, whether they be crossover or whatever, if you're going to do that, because a lot of people will say, oh yeah, my kid improves on that. But the other thing I'd like to emphasize is that, you know, it seems to me we would like to do all of these different screens, all these different approaches. We have to have a biomarker. But since we don't, the only thing at the moment we have, I guess, would be the report from the parents. And I would just advocate for taking all of those patients who appear to be active site mutations or KM mutants and try either GLcNAc or glucosamine. And have the parents on board to be able to record something, and Andy would be a good person to sort of list the clinical things that you might look for. And I don't know the FDA's position at the moment on that, but I do know that GLcNAc has been through phase 1 trials. So that would be something that you could start on and would take a long time, and the FDA should not be terribly opposed to that.
Well, perfectly, Andy has the microphone, so.
Just
go, Andy.
First, one step back from that, just trying to synthesize everything I'm hearing, and I think what I'm hearing is, you know, there's all these different approaches, and some of them can be done in parallel, and others, there's certain steps that need to be completed before you can move on to the others. We've, you know, certainly the natural history study can be done now. Some of the opportunities through the natural history study include collection of biospecimens and storage of those so they can be gone back to, later on when you have another biomarker to look at. Defining the genetics of what's going on with these different mutations, and are some of them gain of function, some of them are loss of function. Trying to parse those out may also be helpful to understand really basic aspects of the disease to be able to move forward in some of the model organisms or in the cell studies and things like that as you move forward.
So one thing I don't want to mislead people, and I think Dan would agree with me, for individual variants, I can see problems, right? Like we have mutations that are unstable, right? That produce an unstable protein. We have ones that clearly are KM mutants for the sugar nucleotide. We have ones that Naomi's not quite ready to publish yet, but that probably don't interact with certain proteins, right? That are TPR variants. So we have individual markers. What we don't have is a global, like we can go, hey, if you're looking at an L254F patient, the thing to look at would be to see if you recover OGA levels. Because OGA levels are at about 8%, right, in the fibroblasts, right? Do they come back, right? So you'd have a marker there, but that wouldn't apply to, Y835C, which is a stability problem for OGT, right? So,
and some of that may be a question of, are these actually different diseases?
Right.
Are their mechanisms different? A gain of function disease versus a loss of function disease, and are your different variants, specific interactions that they're losing in very specific ones, and for your loss of function, common in the glycosylation field is certainly hypomorphic mutations. So is it more a tuning of the amount of flux that's going through these pathways, and that's what's throwing things off? So, defining all of those.
I would argue almost every single one is a hypomorphic mutation. Would you agree with that?
So for all of the mutations that cause a decrease in OGA, there's a test available right now, right? Because for all of the drugs that have been put into people for looking at neurodegenerative diseases, there are the in vivo PET scan, fluorescent sensors.
Yeah, David
they're not
Bacallo's
really
lab
fluorescent,
developed,
right?
Yeah.
But those should really be tested in the animal models, right, where you think you have a decrease in OGA abundance, because you could look whole body then.
Yeah, there's a great O-GlcNAc tau that's been used in PET scanning now, right? That's something that could go in.
But the question is whether that's going to translate from fibroblasts to animal models, because I'm remembering one of Dan's recent papers where when you started to differentiate the cells, you got different phenotypes with your different mutants. But that should be, they should be tools that we're borrowing from the neurodegenerative field already, because some of those, they're putting them into patients already, right?
Right. Yeah, I mean, I think, the one thing that at least is clear to me is there's still a lot of basic research. O-GlcNAc, or OGT and OGA are expressed in every cell in the human body. We have a specific, and that mutation is everywhere in every cell of a patient. And yet the only place we're seeing a phenotype is neurologically, right? And clinodactylism, maybe some hypospadia. I mean, obviously these are big problems if you have them, but the main thing is a neural phenotype. Why? Right? Why just a neural phenotype? It's just the brain's more sensitive? What's going on, right? Because we know O-GlcNAc plays important roles in almost every tissue in our body, and yet we're only seeing phenotypes in the brain, right? Or neurological phenotypes that may or may not be from the brain. Judy?
So I wanted to sort of cautionary tale about for those people who are using model organisms. So some model organisms tend to be outbred. So for instance, we use rats and we go to great pains to make sure that the genetic background is outbred so that the mutation that we want is is there or not there, but the genetic background is not uniform. Most mouse models are in a specific inbred strain of mice, and I think that's true for a lot of model organisms. And so if you put your mutation, the literature is, is rife with examples of, well, we put this mutation in a black 6 and we got this phenotype, but if we put it in a different, background, we might get a different outcome. And we humans, we tend to be outbred. So I think it's really important that if you're using a model organism that is inbred, don't just use one background, use more than one background, just to make sure that the phenotype that you want to attribute to that mutation is true even in a different genetic background.
Sounds like GLcNAc, sounds like all the mutations, all the variants are causing total reduction in O-GlcNAc allelation. Is that true?
No.
Or some reduction of the sum, but we don't know, it could be different for individual target proteins?
Yeah, I mean, I would say for most of the TPR variants that don't affect stability of the protein, some of them affect stability of the protein. If they don't affect stability of the protein, as far as we can tell, again, down to only a depth of maybe 700-800 sites out of the 8,000 sites, there's no defect, right? The O-GlcNAc level. And if you isolate that protein and do rigorous kinetics, right? KM, Vmax, they're fine. They're totally fine with both peptide substrates as well as protein substrates. And I mean, Dan's got mutants like that for sure. I definitely have, they're not ours, right? He's studied variants like that. I've studied variants like that. It's just, there's no change in their O-GlcNAc levels, right?
And that's more based on bulk measurements, but maybe adding subcellular resolution may be a really good way
absolutely,
to, especially
right? I mean, maybe
for
they're
neurons.
all in the mitochondria, right?
Yeah.
Right?
Or we can say like neuronal growth cone dynamics are different. That completely changes the neurodevelopment and then rewire the nervous system differently.
Yeah, I mean, early development, at least to my lab, is something we've thought a lot about because why are the unaffected females skewing like crazy?
Yeah.
They are massively skewed. So is that because of some early development thing, right? Like, what's going on? Why are the mothers so skewed? Yeah. We believe they're skewed towards wild type, but there's only been one family that's been shown. Getting skewing data is easy. Figuring out which way it's skewed is sometimes pretty hard to do.
I mean, so you might be getting ascertainment bias.
Right.
If it wasn't skewed, they wouldn't be asymptomatic and they wouldn't have had a child. And
Right.
But it's also possible that, I mean, I think if I remember back to how exon activation works,
right.
The exist transcript, and I think you're the one who was showing us that there are multiple functions that OGT does, and one of them may have a impact on transcription.
Oh yeah, exist is John Hanover.
Please, yeah, I mean, I'm just wondering if maybe it's affecting the exist, and that's.
Yeah, it was so, actually Stephanie did a lot of work on this when she was in the lab, but we studied a group of patients, human patients with Turner syndrome that have single X chromosomes, either parentally or maternal, either derived from the father or the mother. Very different phenotype. OGT is X imprinted in humans. And so my fear with all of this is that we're looking at a semi-penetrant skewing of exon activation, which is giving rise to lots of mosaicism in terms of the overall phenotype. So, and Stephanie did a very nice piece of work on that, but we haven't gone back to it. We know, for example, OGT and C. elegans, where Barbara Myers looked at exon activation in great detail. There we know quite a bit more about it. And we know that O-GlcNAc is right at the exon activation center. So this thing is evolutionarily tied to sex chromosome compensation. And I think we should follow up as a community on that.
Yeah, and it probably explains why, for such a clear, if you look at L254F, right, it takes like 2 seconds if you're a geneticist, you're like, X-linked disease, right? It's super clear. But if you actually take all of the patients, there's too many females. There's too many female variants, right? It should be extremely rare to see a female variant, and it's not it's uncommon, but I wouldn't call it extremely rare. It's probably, what, 8% of our patients are female, right? That seems. Right, exactly. Right.
Yeah, is there skewing of the de novo females? Sorry, Andrew Edmondson, chop. I only have de novo male patients, so, but for the females, is there like skewing? Have people looked at skewing in affected females? And
I can comment. So we have a Victoria, she's de novo, her skewing is 90 to 10. There are two other twins described, their skewing is 98 to 2. And I don't know, Miranda, if you don't mind, I can share. So parents who are carriers did skewing, and their skewing is 90 to 10, just like our Victoria's.
We don't know which way the skewing is, though.
Yeah.
Right? That's not random, yeah.
So the first catalytic site mutant that was reported was in our 2019 PNAS paper. That's a monozygotic female twin that has 97 to 3% skewing, but we don't know whether it's towards the healthy or the mutated copy. When we make that same mutation in a mouse, out of the 3 mice that we have, these are by far the sickest.
I just want.
And that's a male mouse?
That's a male mouse, yeah,
So
and
so
of course,
compensation.
of course, you know, it's never been demonstrated that that variant can exist in a male, in a human. So, but interestingly, that one is by far the sickest out of our 3 mouse lines. Yeah.
That would make sense.
Just maybe to add more on this X inactivation stuff. So what we have noticed is that, and I think a couple of people in the field have seen this, we've noticed escape from X inactivation as well of OGT. We as a lab are seeing it really when there's a stressful environment in the placenta, for example. So it's possible that we're seeing, I think, phenotype in female because for whatever reason, it might be full skewing, but OGT might escape this X inactivation pattern for a known reason for now. But
and of course, we're doing all this in white blood cells, right? That's where we're looking at X-linked skewing, right? Like, we're not looking at neurons or any other tissues.
Just one last comment from the peanut gallery. The other thing about X inactivation is the X inactivation aficionados, such as Jeannie Lee, Marissa Bartolome, have both called me at various times saying, what is this OGT protein? Because we're seeing developmentally regulated skewing at the OGT locus, not just because it near exists, we're a long non-coding RNA, but because it appears to be an autoregulatory loop. So I think that we ought to put that on a priority list, to at least to think about, particularly to explain the mosaicism. Not your part, but okay.
So if possible, I'd like to spend the last 2 or 3 minutes just talking about ways forward, right? So there are many of us in the field who I think probably want to work together and get something done, right? And we also, right, we have to, it sounds like there might be this FDA thing that seems like it's worth pursuing, figure out how we can work together across international boundaries, which I find very attractive. But there's also, we know there's going to be things that are just going to be like program, project, grant, NIH. And the nice thing is, if we're all talking to each other, those can be 2 totally different things. Like, this is doing basic science, this is making mouse models, this is like, I think the one thing that's clear from this meeting is there's a lot to be done still. And so I think it's going to be really easy to build teams and build groups that are staying off each other's toes as much as possible. And to be honest, 2 people doing the same thing is a really good way of making you believe that it's real, right? like you know, that's just reality. So one of the things I think that would be helpful is I'm going to try to draft after SFG, because I have a small role to play there. But I have a few emails to answer tonight. So, but after SFG, I'm going to try to write up a white paper and share it with everyone who's at this meeting for comment and just to kind of get us thinking. And then hopefully we can just kind of naturally break into groups to think about doing things like going after model organisms and proposing all these various screens, right? Because it wouldn't be that hard to take Drosophila and make 25 variants, right? I mean, that's doable, right? Or whatever. So any final comments from other people? If not, let Andrey wrap it up. Dan, you want to?
I just wanted to say that even though we are working on many of the things that have been discussed today, in principle, our mice are available, right? Nobody has asked, and we certainly haven't said no.
Awesome.
You know, we've published a small paper. There's a bigger paper, hopefully coming out within the next few months. But
Your paper.
maybe.
But I think she can come postdoc with me. All right. Just kidding.
He's doubling your salary, Franz.
You're bringing the embryos with you.
Yeah,
All right.
yeah. No, but seriously, as long as we can work on what it is that we want to work on, if other people want to work on the same things or different things, that's fine, because
that's how science works, right?
they're published and they should be available.
Yep,
I strongly
absolutely.
feel that.
Great.
So in terms of if there's people that want to collaborate in terms of having tissues or slices, that's fine. If you want to have the sperm, that is possible too, and we're looking at ways of depositing them somewhere. Depositing in jacks actually isn't free, because we have to pay for the maintenance of the line there.
Oh, that's annoying.
There is a European repository where it is free, so we're looking into that. But separately from that, if there's people here that want to work on specific questions, come and talk to us, and we can either give you stuff to work with collaboratively, or we can give you the sperm. You can make your own mice, and we'll give you a couple of tips and tricks for breeding, because it's not particularly simple.
Okay.
Yeah.
Yeah, I mean, one of the things I love about the O-GlcNAc field, and it's because Jerry and then John kind of drove this field for a long time, is everybody collaborates, right? Like, if I want anything from Natasha or Stephanie or Michael or whoever in the room, right? I just ask, and it's in the mail the next day usually, right? And so, unless it's Mike, right? It's a little questionable, but that's just me picking on him. But I mean, we all collaborate, we all work together, and right. I mean, one of the great things about having a meeting like this and seeing the families is you remember why you're doing what you're doing, right? It's to try to help people, right? It's right. I think it's Carolyn Bertozzi, right? Being a scientist isn't about being famous. It's not about getting rich. It's about making novel discoveries, and those discoveries outlive you, right? So, comments.
Yeah, quick question. So when we were discussing different models, I believe we didn't discuss mini brains or organoids, right? And the only reason why I know about it, I was asking clinicians, how do we know that GLcNAc is working on Wiki? How do we know that this is working? She said, you need to develop this mini brain. That's all I know. So my question is it something worth exploring? Can it be another potential model?
Yeah, I mean, organoids, I think, are fantastic models. They're coming along. They have limitations, but they also have incredible strengths. I think they're probably going to require specialized laboratories to collaborate with. I mean, I know my lab's not getting, I mean, my lab does a lot of stem cell work, but there's no way we're doing organoids, because I know people who do it for a living. It's a really specialized field, in my opinion, because it, you know, we could probably put our heads together and we can make something that looks like an organoid, but it wouldn't be very rigorous. And these people, that's all they do is make organoids. So I think it would require bringing in some experts, right, if we were going to use that.
Mayo Clinic, Dr. Tan also said that they work with developing organoids and can do that.
Yeah, I mean, if someone's got organoid model, I mean, I'd love to talk to that person, right? I mean, I think probably all of us in this room would love to talk to those people. They're powerful, but they're slow to manipulate, right? I mean, some of the organoid models, at least the ones at the CMM, are 150 day, right? 150 days to make one. Like, so if you want to tweak something, then it's another 150 days, right? So it's really a slow model, but a powerful model.
Do we want?
Go ahead, Andrey. You want to finish this off?
Just before we finish, I think we have just a couple more minutes. the gene therapy, the AAV therapy, do we want to talk about that at all, or it's a little bit too premature at this point?
I mean, if anybody has anything to say about it, I'd love to hear about it. Yeah, Dan.
I would argue there's not much point in doing this until we've shown in a mouse that it helps. And until then, it's a very nice idea, but it's just that, an idea. So we're doing this. Some of the phenotypic studies that we do in mice simply take a very long time, right? You saw the timeline. If we have an idea now and want to try, metabolic intervention, OGA inhibition, we have to make the cohorts, we have to do the assays with large enough cohorts. So basically it just takes, 12 to 18 months to test an idea like AAV or an O-GlcNAc inhibitor. It just takes time.
Right. I mean, I think you're going to have the mouse models, you're going to have the cell models, right? So, I mean, people in my lab have used the OGA inhibitors, they've used glucosamine treatment, they've done overexpress OGT to recover normal O-GlcNAc levels in a mutant that doesn't have that. Wow, that's a cell line, right? Like, there's a long way to go, right? So, yeah. I agree. I think it's a little premature.
I would say the first step there again would be going to like the mouse model to try the AAV and say, does overexpression actually cause problems? It's sort of like one of the questions I'm hearing. So before you invest too much in trying to move that way in humans, you want to make sure that it's not going to cause problems. And the mouse is a good way to do that, better than doing it in cells.
Yeah. Any last comments or questions from anyone? If not, I'm going to let Andrey wrap us up. And thank you all for being here today.
Thank you, Lance. I just wanted to thank all of you guys for really donating your time. It's Saturday. You came here. Some of you are with families. Some of you like away from your families. And as a parent, I deeply, deeply appreciate it. I think that something magical is happening in this room really right now. This is the first ever event dedicated to OGT-XLID. And there are tens of thousands of gene therapies. And what matters for us is that we're creating a community around that. We're creating people who are bounded together by this cause. Now we're going to remember this event. It's going to be our starting point, even though the starting point happened, 30 years ago? With the discovery of Jerry Hart. And hopefully we'll continue to do that on a regular basis. And we'll continue to support each other, to check in how things are going. Maybe we'll set up some regular meetings like over Zoom or over like virtual meetings. There's going to be O-GlcNAc conference early next year, organized by Stephanie, where it's was that? And Mike, sorry, Mike. Yeah, of course. Yeah, and Mike. Yeah. And Mike. Second time. And hopefully we'll have more some developments from this meeting as well, right? It's not a lot of time, but well, we can move quickly probably. Just again, thank you for coming, and to make it even a better community, there are drinks outside of this room over there, so that we have something to remember other than just this meeting. So thank you so much, and see you all hopefully soon, and maybe in half a year, or some of you in a week from now on Zoom calls. Thank you.